In the stillness of the night, when the world seems to slow and the air carries a softer rhythm, it can be calming to think about the quiet foundations of life. Beneath every living thing, beneath every tree, every bird, every ripple of water, and every human breath, there is chemistry. It is not the dramatic kind of chemistry filled with fire and explosions, but a gentler, subtler kind, the chemistry of life. This chemistry does not shout, it does not rush, it unfolds slowly, with precision and harmony, creating the conditions that allow living things to exist and endure. In this sense, chemistry can be thought of as a language, an invisible script, written in molecules and bonds, that all of life reads and follows, without ever needing to be aware of it. To understand this quiet language, it helps first to think about the smallest building blocks of matter. Everything we can touch, everything we can see, everything we can feel, is made of atoms. Atoms themselves are unimaginably small, so small that a single grain of sand contains more of them than there are stars in the entire night sky. Each atom carries within it a tiny nucleus made of protons and neutrons, around which electrons drift in patterns of motion. On their own, atoms are simply fragments of possibility, but when they come together, when they connect and share, they form molecules. And molecules are the beginning of life's story. They are the words and sentences in the language of chemistry. Among the countless atoms in the universe, only a few have taken center stage in the making of living beings. Carbon, hydrogen, oxygen, and nitrogen. These four in particular are the familiar characters, forming about 96% of the mass of a human body. They appear so often because they can combine in ways that are stable and yet endlessly diverse. Carbon, for instance, has a rare gift. It can form four strong connections at once, linking into chains, rings, and branching structures that are the skeletons of life's molecules. Hydrogen and oxygen come together to create water, the universal solvent, without which life, as we know it, would be impossible. Nitrogen forms the backbone of amino acids, the small units that assemble into proteins, and also gives shape to the molecules of DNA and RNA that hold life's instructions. Together, these elements weave a quiet but resilient tapestry, one that is both delicate and enduring. Life's chemistry is not random, though it may appear so at first glance. It follows patterns, guided by the ways atoms interact and by the laws of energy. Bonds form, bonds break, and bonds form again, all in a ceaseless dance that sustains living systems. Within your own body at this very moment, millions upon millions of chemical reactions are unfolding silently. Molecules of glucose are being broken down to release energy. Chains of amino acids are being folded into proteins that will mend tissues, carry signals, and support growth. Water molecules are moving into and out of cells, keeping balance in place. None of this can be felt directly, and yet it is happening with exquisite order. Chemistry also holds within it a sense of duality, simplicity on one side, complexity on the other. The rules themselves are few and elegant. Atoms strive to achieve stability. Molecules form according to valence and charge. Reactions shift depending on the energy available. And yet from these simple rules arises staggering diversity. A handful of elements and a small set of bonds give rise to forests, oceans, coral reefs, and the beating of your heart. Life, in all its forms, is proof of how complexity can emerge from simplicity, how quiet rules can lead to wondrous outcomes. In the history of our planet, chemistry was present long before life appeared. For billions of years, Earth was a restless sphere of rock, gas, and water. Volcanoes erupted, oceans gathered, and lightning struck the atmosphere. Out of this environment came the earliest reactions that stitched together organic molecules. Carbon-based compounds, that are the precursors to life. Amino acids, formed in pools of water, sugars assembled from simpler molecules. Fatty acids align themselves into thin membranes, shaping the first fragile boundaries of primitive cells. Slowly, patiently, chemistry laid the groundwork for biology. The spark of life was not a single dramatic event, but a gradual unfolding, a soft layering of possibilities, each step supported by chemical bonds that proved just strong enough to hold. Thinking about chemistry in this way can be comforting. It reminds us that life is not precarious, though it sometimes feels so. It is anchored in fundamental processes that have been steady for billions of years. The same hydrogen bonds that help hold together your DNA are the same kind of bonds that existed when Earth's first molecules were forming in ancient seas. The same oxygen atom you breathe tonight may have once been part of a plant leaf, or a cloud, or even part of a star that burned long before the Earth was formed. Chemistry connects us not only to all living beings around us, but also to the deep history of the cosmos. When we view chemistry through the lens of life, it becomes less about formulas and more about relationships. Atoms do not exist in isolation, but join together in endless combinations. Molecules are not static, but shift, rotate, and transform. Reactions are not chaotic, but guided, often helped along by enzymes that act as caretakers of speed and precision. In every drop of blood, in every cell, these relationships play out quietly, ensuring that energy is available, that signals are sent, that balance is kept. Life is nothing more and nothing less than chemistry arranged in a way that can renew itself, sustain itself, and grow. As you settle into rest, you may imagine these processes unfolding inside you, a quiet orchestration that requires no effort on your part. Your body carries within it trillions of cells, and within each cell countless molecules move in harmony. There is no need to direct them. They follow the language of chemistry, and they have always done so. This silent order continues even as you drift into sleep, ensuring that when you awaken, life's chemistry will have carried you gently through the night. Thus, the introduction to life chemistry is not a lesson to be memorized, but a reminder to be felt. It is the reassurance that at the heart of living beings lies a deep and timeless simplicity. Atoms, molecules, and bonds, quiet, patient, and reliable, form the invisible foundation upon which all of existence rests. From the smallest bacterium to the largest tree, from the faintest heartbeat to the brightest star, chemistry is the thread that ties it all together. And in this moment, as you listen in calm stillness, you are part of that great and gentle weave. 2. The elements of life. When we look at the vastness of the natural world, at the forests, the oceans, and the skies above, it is easy to be overwhelmed by the richness and variety of living things. Yet beneath all of this diversity lies a surprising simplicity. The forms of life we see around us are built from a limited set of ingredients, a handful of chemical elements that come together again and again in countless combinations. These elements are not rare or exotic. They are familiar, woven into every breath we take, every sip of water we drink, and every bite of food we eat. To understand the basics of life chemistry, it helps to begin with these essential elements, for they are the alphabet from which the sentences of life are written. If we could take the human body, or any living body, and break it down into its elemental composition, we would find that just 4 elements dominate above all others. These are carbon, hydrogen, oxygen, and nitrogen. They make up more than 96% of the mass of our bodies. Around them gather smaller but still important contributors like phosphorus, sulfur, calcium, potassium, and a collection of trace elements present only in tiny amounts, yet no less essential. Each of these elements carries its own story, its own subtle role, and together they create the chemistry that sustains life. Oxygen, the most abundant element in the human body, is present in every breath. It makes up much of the water within us, and water itself accounts for the majority of our weight. Oxygen is also found in carbohydrates, fats, and proteins, the molecules that store and provide energy. In its gaseous form, oxygen is inhaled with each breath and carried by the bloodstream to every cell, where it participates in cellular respiration. This quiet process allows the energy stored in food molecules to be released and transformed into usable forms. Without oxygen, life as we know it would falter almost instantly, reminding us that this simple element is one of life's most constant companions. Hydrogen, though the smallest and lightest of all atoms, is equally vital. Like oxygen, it is a central component of water, forming bonds that give water its unique and life-sustaining properties. Hydrogen is also threaded through the molecules of carbohydrates, proteins, and lipids, shaping their structures and influencing their functions. Beyond structure, hydrogen plays a role in energy transfer. In the form of protons and electrons, hydrogen participates in the flow of energy within cells, especially during processes that generate ATP, the energy currency of life. It is remarkable to consider that the simplest of elements, containing only a single proton and electron, is so deeply woven into the complexity of every living system. Carbon stands out as the true backbone of life's chemistry. Unlike any other element, carbon can form 4 stable covalent bonds at once, creating a wide array of structures. It can link to itself to form chains, branch into complex frameworks, and arrange into rings. This versatility allows for the construction of molecules of astonishing variety and complexity, from the smallest sugars to the largest proteins and strands of DNA. Because of its flexibility, carbon is sometimes called the element of life, and rightly so. Without carbon, the architecture of living systems would collapse. Its ability to form stable yet dynamic structures gives rise to the diversity of life we see today. And it is no exaggeration to say that carbon is the silent architect behind all living forms. Nitrogen, while less abundant than oxygen, hydrogen, or carbon, is no less important. It is found in every amino acid, the small building blocks that link together to form proteins. Proteins, in turn, carry out the vast majority of tasks in living organisms, from catalyzing reactions to building tissues to transmitting signals. Nitrogen is also a central part of nucleic acids, including DNA and RNA, which carry the genetic instructions of life. Every thought, every movement, every inherited trait rests on the presence of nitrogen in these molecules. Without nitrogen, proteins could not exist, and without proteins, life itself would come undone. Beyond these 4 elements, others contribute in subtler but equally essential ways. Phosphorus, for example, is part of the backbone of DNA and RNA, linking nucleotides into long chains that can store and transmit information. It is also a key component of ATP, the molecule that serves as the cell's main energy carrier. Sulfur appears in certain amino acids and helps stabilize protein structures through disulfide bonds, small links that add strength to complex molecules. Calcium is widely recognized for its role in building bones and teeth, but it also acts as a signaling molecule inside cells, helping to regulate processes as diverse as muscle contraction and neurotransmission. Potassium and sodium maintain electrical balance across cell membranes, allowing nerves to fire and muscles to move in smooth coordination. Then there are the trace elements, required in amounts so small they are measured in milligrams or even micrograms. Yet, despite their scarcity, they are indispensable. Iron, for instance, binds oxygen in hemoglobin, enabling red blood cells to carry it throughout the body. Zinc contributes to the function of enzymes that guide chemical reactions. Iodine is essential for the production of thyroid hormones, which regulate metabolism. Copper, manganese, selenium, and others each play quiet but crucial roles, ensuring that life's delicate machinery runs without interruption. Though present in small amounts, these elements illustrate how even the tiniest contributors can be vital to the whole. It is worth noting that these elements are not unique to humans or even to Earth. They are among the most common in the universe, forged in the cores of stars and scattered across space through cosmic events. The carbon in your body may once have been part of a distant star. The oxygen you breathe may have drifted through countless forms before entering your lungs tonight. In this sense, the elements of life connect us not only to other living beings, but also to the cosmos itself. We are, in the quietest and most literal way, made of stardust, assembled into forms capable of thought, feeling, and rest. The fact that life relies on such a small group of elements also speaks to a kind of elegance. Nature has found a way to build staggering diversity with only a limited palette, from a single seed to the vast canopy of a rainforest, from the smallest bacterium to the blue whale. All are variations on the same chemical theme. The difference lies not in the elements themselves, but in how they are arranged, in the patterns of bonds and structures that emerge from them. This is chemistry's gentle secret. Infinite possibility contained within finite means. As you reflect on these elements, you may find comfort in their constancy. They are always present, always quietly working, regardless of our awareness. The breath you just took drew in oxygen. The water you drink replenishes hydrogen and oxygen alike. The food you eat provides carbon and nitrogen, fueling cells and building tissues. At every moment, these elements move through you, maintaining life in ways too subtle to feel, but too essential to ignore. Even as you rest, they continue their work, forming bonds, breaking bonds, and sustaining balance. To know the elements of life is to glimpse the foundation upon which everything else rests. They are not complicated, nor are they inaccessible. They are simple, stable, and enduring, the quiet companions of existence. And as you drift deeper into stillness, you may take ease in knowing that these elements are within you, around you, and have always been part of the story of life, patiently carrying out their roles in the great chemistry of being. 3. Water, the universal solvent. If there is one substance that life cannot do without, it is water. Every living organism, from the smallest bacterium to the tallest tree, is built upon the presence and availability of water. Our own bodies, when measured by weight, are composed of more water than anything else, often around 60%. Cells themselves, the tiny compartments that form the foundation of life, are largely watery spaces, filled with and surrounded by this simple but remarkable liquid. Water is not merely a backdrop for life, not just a medium in which other processes unfold. It is an active participant in nearly every chemical reaction and biological process. To understand the basics of life chemistry, it is natural to pause and consider water, the quiet, universal solvent that makes life possible. At first glance, water seems ordinary. It falls from the sky as rain. It flows in rivers. It gathers in oceans. And it sits in glasses beside us. But in its simplicity lies a collection of unusual properties that set it apart from most other substances. Each molecule of water consists of 2 hydrogen atoms bound to 1 oxygen atom, arranged in a bent shape rather than a straight line. This geometry, combined with differences in electrical charge between the oxygen and hydrogen atoms, makes the molecule polar. In other words, one side of the molecule carries a slight negative charge, while the other side carries a slight positive charge. This polarity is the secret behind water's unusual behavior and its extraordinary role in supporting life. Because of this polarity, water molecules form hydrogen bonds with one another. These bonds are weaker than the covalent bonds that hold atoms together within a single molecule, yet strong enough in their collective presence to give water cohesion, stability, and structure. Hydrogen bonding allows water molecules to cling together, creating surface tension that permits small insects to walk across ponds. It gives water a relatively high boiling point and melting point compared to other molecules of similar size, ensuring that liquid water exists across the temperature ranges found on Earth. Without hydrogen bonding, water would behave more like a gas at normal temperatures, and life as we know it could never have taken root. One of water's most life-sustaining properties is its ability to dissolve a wide variety of substances. Because it is polar, it can interact with many kinds of charged or polar molecules, pulling them apart and surrounding them with a shell of water molecules. This is why salts, sugars, and so many other compounds dissolve easily in water. Within living organisms, this property allows nutrients, gases, and waste products to be carried in solution. Blood, for instance, is mostly water, carrying oxygen, glucose, salts, hormones, and countless other substances to where they are needed. Inside cells, the cytoplasm is a watery solution in which enzymes, proteins, and organelles carry out their work. Water, by dissolving and transporting these molecules, ensures that the chemistry of life can take place smoothly and continuously. Water also plays a crucial role in regulating temperature. Because hydrogen bonds absorb and release heat slowly, water resists rapid changes in temperature. This high specific heat capacity means that large bodies of water, such as oceans and lakes, can absorb enormous amounts of energy without becoming dramatically hotter or colder. On a global scale, this moderates Earth's climate, preventing extremes of heat and cold that would be hostile to life. On a smaller scale, within living organisms, water helps maintain stable internal temperatures. The blood and fluids circulating in our bodies absorb excess heat and distribute it evenly, preventing dangerous fluctuations. When we perspire, the evaporation of water from the skin carries away heat, cooling the body with quiet efficiency. This thermal stability is one of the many reasons water is indispensable to living systems. Another unusual property of water is that it expands when it freezes. Most substances become denser as they solidify, but water behaves differently. When it freezes, the hydrogen bonds lock the molecules into a rigid lattice that takes up more space than liquid water. This is why ice floats, though it may seem like a simple curiosity. This property is vital for life on Earth. In lakes, ponds, and oceans, ice forms on the surface, creating a layer of insulation that protects the liquid water below from freezing solid. This allows fish and other aquatic organisms to survive even through the harshest winters. Without this anomaly, bodies of water might freeze from the bottom up, destroying ecosystems and making survival impossible. Water is also deeply involved in the chemical reactions of life. In metabolism, many reactions involve either the removal of water to join molecules together or the addition of water to break them apart. The formation of proteins, carbohydrates, and lipids often requires dehydration reactions, where a molecule of water is released as bonds are formed. Conversely, in digestion and other processes, hydrolysis reactions use water to split large molecules into smaller, more manageable pieces. Beyond these reactions, water itself participates directly in photosynthesis, where plants split water molecules to release oxygen and generate the electrons needed to capture sunlight's energy. In respiration, water is formed as a final product when electrons flow to oxygen at the end of the energy harvesting chain. In these ways, water is not just a solvent but a partner in the flow of energy through living systems. Even at the cellular level, Water shapes the very structure of biological molecules. Proteins fold into their functional shapes, largely because of the way they interact with water. Portions of proteins that are attracted to water, called hydrophilic regions, arrange themselves on the outside, while water-repelling, or hydrophobic, regions fold inward, away from contact with water. Similarly, the membranes that surround cells are built from lipids that organize themselves into bilayers, with hydrophobic tails tucked inside and hydrophilic heads facing outward toward water. This self-assembly, driven by interactions with water, provides the barrier that separates the cell's internal world from the environment around it. In thinking about water, one can also appreciate its quiet ubiquity. It cycles endlessly through the planet, moving from clouds to rain, from rivers to oceans, from soil into plants, and back again. This water cycle sustains ecosystems, redistributing nutrients and supporting life in every environment. In your own body, water cycles too. It enters when you drink and eat. It circulates through blood and lymph, and it leaves again through breath, sweat, and elimination. Yet the molecules of water themselves remain the same, circulating through countless lives, countless systems, across the ages. A glass of water you drink tonight may contain molecules that once passed through an ancient forest, or an ocean current, or even a prehistoric creature. There is comfort in knowing that something so simple and abundant is also so indispensable. Water does not demand attention, yet it underlies every moment of existence. It cushions joints, moistens tissues, carries nutrients, and flushes wastes. It helps shape climates, erodes mountains, and nourishes valleys. It is patient, cycling endlessly without ever being consumed. In every way, water is the quiet companion of life, the substance in which all of chemistry's delicate balances are carried out. As you rest and breathe calmly, you may imagine the water within you, moving gently from cell to cell, carrying messages, dissolving nutrients, balancing temperature, and maintaining harmony. Without conscious effort, it flows through you, part of the same cycle that has moved across Earth for billions of years. To understand water is to recognize that life depends not on complexity alone, but on the reliable presence of a simple molecule, formed from 2 atoms of hydrogen and 1 of oxygen, bound together in quiet resilience. It is the universal solvent, the gentle medium of existence, and it continues its work even as you drift into sleep. 4. The backbone of living chemistry. Among all the elements in the universe, there is one that stands out as uniquely suited to sustain life in its immense variety and complexity. That element is carbon. Quiet and unassuming, carbon is not flashy like gold, nor heavy like iron, nor glowing like neon. Yet it has a quality that makes it unmatched: versatility. Carbon can form an extraordinary number of connections, building structures as simple as a single chain and as elaborate as the double helix of DNA. It is because of this adaptability that carbon is often called the backbone of living chemistry, and with good reason. Without carbon's flexibility, the diversity of life we see around us would not be possible. At the heart of carbon's importance lies its atomic structure. Each atom of carbon contains 6 protons, 6 neutrons, and 6 electrons. The electrons are arranged in such a way that 4 of them are available to form bonds with other atoms. This ability to make 4 covalent bonds at once gives carbon a unique place among the elements. While hydrogen can form only 1 bond, and oxygen 2, and nitrogen 3, carbon can branch out in 4 directions. This single fact is what allows carbon to serve as the foundation for molecules of almost limitless complexity. Chains, rings, branches, and three-dimensional frameworks all become possible because carbon can connect in so many ways. When carbon atoms bond with one another, they can create long chains that serve as the skeletons of molecules. These chains can be straight, unbroken lines, or they can branch and fold back upon themselves. In addition to chains, carbon atoms can also form rings, which provide stability and allow for the creation of aromatic compounds with distinct properties. Some carbon structures are small, like methane, which has just one carbon bonded to four hydrogens. Others are vast, like the large polymers that make up proteins, carbohydrates, and nucleic acids. At every scale, carbon's ability to link in multiple directions provides a scaffold upon which life's chemistry is built. Another quality of carbon is its ability to form bonds of varying strength and character. Carbon can create single bonds, where two atoms share one pair of electrons. It can also form double bonds, where two pairs are shared, or triple bonds, where three pairs are exchanged. Each type of bond alters the shape and behavior of the molecule. Single bonds allow rotation, giving molecules flexibility. Double bonds restrict rotation, locking structures into particular shapes. Triple bonds bring even greater strength and rigidity. By mixing these types of bonds, carbon-based molecules can be soft or strong, flexible or stiff, stable or reactive. This diversity allows for the precise tailoring of molecules to perform specific roles in living organisms. Consider the molecules that serve as the cornerstones of life. Carbohydrates, for instance, are chains and rings of carbon atoms, decorated with hydrogen and oxygen. These molecules provide energy to cells, storing the energy of the sun captured by plants. Lipids, another class of carbon-based molecules, consist of long carbon chains that form fats and oils. These molecules store energy for the long term and create the membranes that surround cells. Proteins, built from amino acids, are carbon frameworks that fold into intricate shapes, performing tasks as varied as catalyzing reactions, transporting substances, and forming structural tissues. Nucleic acids, such as DNA and RNA, are long chains of carbon-containing nucleotides, storing and transmitting genetic information. Each of these essential macromolecules owes its existence to carbon's versatility. Carbon also interacts easily with other elements, particularly hydrogen, oxygen, nitrogen, phosphorus, and sulfur. This flexibility allows for the creation of molecules with distinct chemical personalities. Some molecules are hydrophilic, meaning they interact well with water, while others are hydrophobic, repelling water. Some carry charges, while others remain neutral. These subtle variations enable molecules to form the complex networks necessary for metabolism, signaling, and regulation within living systems. It is this interplay carbon linking with diverse partners that gives life its intricate choreography. An especially striking example of carbon's role is in the structure of DNA. Each strand of DNA is composed of a sugar-phosphate backbone, where the sugar is a carbon-based molecule. Attached to these sugars are nitrogenous bases, themselves carbon-rich structures. The specific pairing of these bases encodes the genetic instructions of life, the sequences that guide the formation of proteins, and ultimately shape every living being. Without carbon's capacity to form stable backbones and versatile side groups, the double helix could not exist, and heredity as we know it would be impossible. Carbon is also central to the flow of energy in living systems. Through photosynthesis, plants capture carbon dioxide from the atmosphere and, with the help of sunlight, transform it into glucose and other carbohydrates. These molecules then serve as fuel not only for the plants themselves but also for animals, fungi, and countless microorganisms that consume them. Through respiration, glucose is broken down, releasing the stored energy to drive cellular processes. The cycle of carbon from atmosphere to plant to animal and back to atmosphere is one of the most fundamental processes on Earth. This carbon cycle ensures that energy moves smoothly through ecosystems, sustaining life at every level. Beyond biology, carbon shows its versatility in countless other forms. Pure carbon can appear as diamond, one of the hardest substances known, where each atom is tightly bonded to four others in a rigid lattice. It can also appear as graphite, soft and slippery, where carbon atoms are arranged in flat sheets that slide past one another. In recent decades, scientists have discovered new carbon structures, such as fullerenes and nanotubes, where atoms form hollow spheres or long, cylindrical tubes with remarkable properties. These examples remind us that carbon is not only central to life but also endlessly adaptable in the physical world. What makes carbon even more remarkable is its abundance. It is one of the most common elements in the universe, produced in the hearts of stars through the process of fusion. When those stars reached the ends of their lives and exploded as supernovae, carbon was scattered into space, seeding planets, and eventually becoming the foundation of living organisms. In this sense, the carbon within your body was forged billions of years ago in distant stars. Every breath you take, every movement you make, depends on atoms that carry within them the history of the cosmos. To reflect on carbon is to recognize that life is not separate from the universe but deeply connected to its very fabric. As you rest, it may be soothing to imagine the quiet work of carbon within you. Long chains of carbon atoms form the sugars that fuel your cells. Folded carbon frameworks make up the proteins that keep your heart beating and your muscles moving. The spiral staircase of DNA in every cell is built upon carbon's sturdy backbone, carrying instructions that have been passed down across countless generations. Carbon is everywhere in you, silent and unassuming, holding together the molecules that make you who you are. There is a quiet beauty in this realization. From the smallest seedling to the tallest tree, from the simplest bacterium to the complexity of human thought, all are variations on carbon's theme. It is the common thread that unites the diversity of life, the backbone that makes the chemistry of living possible. And even as you drift into sleep, carbon continues its work, bonds forming and breaking, energy flowing, molecules assembling, all guided by the quiet strength of this extraordinary element. 5. Molecular bonds and gentle interactions. In the quiet chemistry of life, everything rests on connections. Atoms do not simply float alone in endless space. Content in their solitude, they reach out, form attachments, and in those attachments, new possibilities emerge. These connections are called bonds, and they are the invisible threads that weave atoms into molecules, and molecules into the structures that sustain living systems. To reflect on these bonds is to see how something as delicate and unseen as the sharing of electrons can shape forests, oceans, bodies, and minds. Bonds and interactions are the quiet architects of life, guiding how molecules come together, how they hold, and how they separate again when the time is right. The strongest and most foundational of these connections are covalent bonds. A covalent bond is formed when two atoms share a pair of electrons. This simple act of sharing creates stability, because atoms seek to complete their outer electron shells, and by joining forces, they achieve a more balanced state. The covalent bond is not a rigid clamp but more like a gentle embrace, holding atoms together with just the right firmness. In water, for instance, oxygen shares electrons with two hydrogen atoms, creating a molecule that is both stable and essential. In glucose, carbon atoms link through covalent bonds to form the backbone of energy molecules. Proteins, too, are built on long chains of covalently bonded atoms, folded into intricate shapes that carry out countless functions. Everywhere in life, covalent bonds are the foundation, ensuring that molecules remain whole and purposeful. Not all covalent bonds are identical. Sometimes the sharing of electrons is equal, but often it is not. When one atom holds the electrons closer, the bond becomes polar, creating regions of slight positive and negative charge. Water is a perfect example. Oxygen, being more electronegative, pulls electrons closer to itself, leaving the hydrogen slightly positive. This unequal sharing gives water its polarity, which in turn makes it capable of dissolving so many other substances. Polarity, though subtle, becomes powerful, shaping interactions between molecules in ways that ripple throughout biology. A polar bond is like a conversation where one voice is stronger, yet the exchange still holds together. Another kind of bond is the ionic bond. Unlike covalent bonds, which involve sharing, ionic bonds involve giving and taking. One atom transfers an electron to another, creating two charged ions, one positive, one negative, that are held together by electrostatic attraction. Salt, or sodium chloride, is a familiar example. Sodium readily gives up an electron, becoming positively charged, while chlorine accepts it, becoming negatively charged. The result is a crystal lattice, held together by countless ionic bonds. In the human body, ionic bonds appear in countless salts that dissolve in water, releasing ions that participate in nerve signaling, muscle contraction, and fluid balance. Though less stable in watery environments than covalent bonds, ionic bonds play a quiet but indispensable role in the chemistry of life. Then there are hydrogen bonds, lighter and more transient than covalent or ionic bonds, yet no less important. A hydrogen bond forms when a hydrogen atom, already attached to a more electronegative atom, is attracted to another electronegative atom nearby. This weak attraction, often no more than a fleeting pull, becomes immensely powerful in numbers. In water, hydrogen bonds link molecules together, giving water its cohesion, its surface tension, and its resistance to rapid temperature changes. In DNA, hydrogen bonds pair the bases across the double helix, holding the two strands together while still allowing them to separate during replication. Proteins, too, rely on hydrogen bonds to fold into their functional shapes, with these delicate interactions stabilizing their twists and turns. Though each hydrogen bond is fragile, together they form a network strong enough to sustain the architecture of life. Alongside these bonds are van der Waals interactions, even weaker than hydrogen bonds, yet pervasive. These occur because electrons within atoms are never perfectly still. At any moment, a slight imbalance may create a temporary dipole, a small region of uneven charge. This fleeting polarity can induce a similar polarity in neighboring atoms, creating a brief attraction. On their own, van der Waals interactions are faint and transient. Yet, when molecules come into close contact, especially in large numbers, these interactions become meaningful. They help proteins fold with precision, assist lipids in forming membranes, and allow geckos to climb walls by harnessing countless tiny attractions between their feet and the surfaces they touch. These quiet forces, nearly invisible, demonstrate that even the most delicate interactions can have profound effects. In the larger picture, these different kinds of bonds and interactions do not act in isolation. They cooperate, overlap, and reinforce one another, creating a web of stability and flexibility. A single protein, for example, is held together by covalent bonds in its backbone, shaped by hydrogen bonds in its folds, guided by ionic bonds between charged groups, and stabilized further by van der Waals forces among its atoms. This combination ensures that the protein is strong enough to endure, but flexible enough to function. Life depends on this balance. Rigid structures would be lifeless, while too much fluidity would be chaotic. It is in the balance of bonds that life finds its harmony. Bonds also carry within them the quiet energy of change. To break a bond requires energy, and to form a bond releases energy. This ebb and flow of energy is the essence of chemical reactions. In respiration, for instance, the covalent bonds in glucose are broken, and in the process energy is released to power the formation of ATP, the cell's energy currency. In photosynthesis, new bonds are formed as carbon dioxide and water are assembled into glucose, capturing the energy of sunlight in chemical form. Every reaction in metabolism is, at its heart, the making and breaking of bonds, the shifting of electrons, the rearranging of atoms into new and purposeful forms. It is remarkable to consider that all of life's richness, every tree, every songbird, every heartbeat, is the outcome of these invisible connections. We cannot see covalent bonds holding molecules together, nor ionic bonds drawing ions close, nor hydrogen bonds weaving molecules into shapes. Yet without them, nothing would exist. Chemistry, through bonds, provides not only structure, but also possibility. It is in these connections that atoms transcend their individuality and become part of something larger, something alive. Even within you now, as you rest, bonds are forming and breaking in endless cycles. Food molecules are being broken apart in your cells, releasing energy. Proteins are folding into shapes, stabilized by hydrogen bonds. DNA is unwinding and replicating, its strands held delicately together by bonds that can break and reform with precision. Water molecules are clinging to one another, forming hydrogen bonds that give your fluid stability. Each of these bonds is silent, invisible, and tireless, sustaining you without any need for awareness or effort. To think of molecular bonds is to think of life as a tapestry woven from threads too fine for the eye to see. And yet, these threads are everywhere, binding the world together. They are gentle, flexible, strong, and subtle. They allow for both permanence and change, for both order and adaptation. They remind us that life's beauty does not come from grand gestures, but from quiet, persistent connections that hold and release in balance. As you drift deeper into calm, you may imagine the molecules within you, connected by bonds of many kinds. Each bond a whisper of stability, each interaction a note in the music of life. Together, they form the harmony that sustains your body, your thoughts, and your very being. The bonds may be unseen, but their effects are everywhere, carrying life forward in silence, in patience, in quiet strength. 6. Simple molecules, profound roles. When we think about the chemistry of life, it is easy to imagine grand and intricate molecules, the spiraling double helix of DNA, the folded shapes of proteins, or the vast membranes of cells. These complex structures are indeed vital, but at the foundation of life's chemistry lie much simpler molecules, small and seemingly unremarkable. These molecules carry out roles so profound that without them, the complexity of life could never be sustained. They are the quiet actors behind the scenes, moving in and out of cells, passing through membranes, and enabling the processes that keep organisms alive. To appreciate the elegance of life's chemistry, it is worth pausing to reflect on these simple molecules and the profound roles they play. The first, and perhaps most familiar of these simple molecules, is oxygen. Essential for the survival of most living organisms, oxygen is a gas that dissolves into blood and diffuses into cells, where it is used in cellular respiration. Within the mitochondria of cells, oxygen serves as the final acceptor of electrons in the chain of reactions that extract energy from food. Without oxygen, the flow of electrons would halt, energy would not be released, and cells would falter. Yet oxygen does not act alone. It is accompanied by carbon dioxide, another simple molecule that carries away the waste of respiration. Carbon dioxide, though often thought of as a byproduct, is no less important. It dissolves in blood, diffuses out through the lungs, and in plants becomes the starting material for photosynthesis, where it is captured and transformed into carbohydrates. The cycle between oxygen and carbon dioxide is a quiet rhythm, sustaining both animals and plants in mutual exchange. Another class of simple molecules with profound roles is the sugars. Glucose, a molecule made of just 6 carbons, 12 hydrogens, and 6 oxygens, is the primary fuel for most cells. It is small enough to travel easily through the bloodstream, and simple enough to be broken down step by step to release energy in a controlled way. In glycolysis, glucose is split into smaller fragments, releasing energy that is captured in the form of ATP, the universal energy currency of cells. That ATP, another relatively small molecule, powers nearly every process in life, from muscle contraction to the synthesis of DNA. It is astonishing to think that the energy that allows you to move, to think, and even to dream as you sleep, is ultimately drawn from the quiet breakdown of glucose molecules within your cells. Water, too, is a simple molecule, composed of only 2 hydrogens and 1 oxygen, yet its roles are countless. It provides the solvent in which most biological reactions occur. It participates directly in hydrolysis reactions, breaking larger molecules into smaller ones. It helps regulate temperature, cushions organs, and allows nutrients and wastes to move freely through the body. Water molecules form the matrix in which ions like sodium, potassium, and calcium drift, each ion essential to the signaling and regulation that keep life balanced. Though water seems ordinary, its presence is indispensable. It is not only the medium of life, but also a participant in the very reactions that sustain life. Another simple but essential molecule is ammonia, NH, or its ionized form, ammonium. Though toxic in large amounts, nitrogen compounds like these serve as building blocks for amino acids and nucleotides. In the natural environment, microorganisms convert nitrogen gas from the atmosphere into ammonia, which plants can then use to synthesize the molecules of life. This process, known as nitrogen fixation, transforms a simple, inert gas into the basis of proteins and DNA. Without these small nitrogen compounds, the biosphere could not renew itself, and life would not endure. Lipids, while often associated with larger structures like membranes, also rely on simple molecules as their building blocks. Fatty acids, long chains of carbon and hydrogen with a carboxyl group at one end, are themselves relatively small. Yet they combine into larger lipids that store energy and form protective barriers around cells. Cholesterol, another simple molecule, is small enough to move within membranes, but vital enough to regulate their fluidity and serve as a precursor to hormones. Hormones themselves, like adrenaline or cortisol, are often small molecules, circulating through the bloodstream and carrying signals that regulate stress, metabolism, and countless other processes. Ions, too, are among the simplest participants in life's chemistry, yet their roles are profound. Sodium, potassium, calcium, chloride, and magnesium are all charged particles dissolved in the watery environment of cells. Their movement across membranes generates electrical impulses that allow neurons to fire and muscles to contract. The balance of these ions determines blood pressure, hydration, and even the rhythm of the heartbeat. Each tiny ion, though simple in form, carries with it the power to regulate systems that span the entire body. Consider also carbon monoxide, nitric oxide, and other tiny gases. Though dangerous in excess, these small molecules also act as messengers in controlled amounts. Nitric oxide, for example, helps blood vessels relax, regulating blood pressure and flow. In the immune system, it participates in defense, destroying pathogens. These gases diffuse freely through membranes, their simplicity granting them access to places that larger molecules cannot reach. Though they are small, their influence is wide. Even the most fundamental processes of genetics rely on simple molecules. Nucleotides, the building blocks of DNA and RNA, are composed of a sugar, a phosphate group, and a nitrogenous base. Each nucleotide is simple, yet when linked together they create the chains that carry genetic information. The pairing of bases adenine with thymine, guanine with cytosine is guided by simple hydrogen bonds, forming the stable yet flexible structure of the double helix. Without these basic molecular interactions, the vast instructions for building and sustaining life could not be stored or passed on. The beauty of these simple molecules lies not only in their roles individually but also in the way they connect into cycles. The oxygen you breathe becomes part of water and carbon dioxide. The glucose you eat is broken down, releasing energy and producing carbon dioxide, which is then taken in by plants and converted back into glucose and oxygen. The nitrogen from simple ammonia molecules cycles through soil, plants, and animals, eventually returning to the atmosphere. These cycles illustrate how life's chemistry is not a collection of isolated events but an interconnected web of exchanges, all revolving around the quiet work of simple molecules. What makes these molecules so profound is that they bridge the gap between simplicity and complexity. They are small enough to move easily, to react quickly, and to connect seamlessly with larger structures. Yet they are significant enough to sustain processes as vast as ecosystems and as intricate as human thought. They remind us that life does not depend solely on grand, complex molecules, but also on the humble, small ones that carry out the most essential tasks. As you rest, it may be soothing to imagine these molecules at work within you. Oxygen molecules entering your lungs and diffusing into your blood. Glucose circulating quietly, ready to fuel the cells. Water flowing into and out of tissues, balancing and cleansing. Ions drifting across membranes, sending signals that keep your heart steady and your mind calm. Each of these molecules is simple in form, yet together they create the symphony of life. Their quiet presence sustains you, night and day, without demand or effort. In the end, the lesson of simple molecules is one of humility and wonder. Life does not rely only on complexity, but finds its strength in the simplest of forms. The air, the water, the nutrients, the ions, all are small, all are quiet. They are the profound foundation upon which all else is built. The first notes in the melody of existence. And as you drift into sleep, these molecules continue their work, unseen yet tireless, guiding the chemistry of life with patience and grace. 7. Macromolecules, the four pillars. When we look at the chemistry of life, we find that it is built upon a foundation that is both simple and profound. At the smallest scale, there are atoms, the individual units of matter. These atoms join to form small molecules such as water, oxygen, or glucose. Yet life requires more than small fragments. It depends on large, intricate structures that can store energy, provide strength, transmit information, and carry out reactions. These larger structures are called macromolecules. The word itself means large molecules, but their importance far outweighs their size. They are the great pillars upon which the house of life is built. Proteins, lipids, carbohydrates, and nucleic acids. These four families of macromolecules support nearly every function of living systems. Each one has its own character, its own role, and together they form the framework of all biological existence. Proteins are perhaps the most versatile of the macromolecules. Built from smaller units called amino acids, proteins fold into complex shapes that determine their function. Some proteins act as enzymes, catalysts that speed up chemical reactions with remarkable precision. Without enzymes, the reactions necessary for life would occur too slowly to sustain us. Other proteins serve as structural materials, forming the fibers of muscles, the strength of connective tissues, and the scaffolding inside cells. Still others act as messengers, carrying signals between cells and across systems. Hemoglobin, a protein in red blood cells, binds oxygen and carries it through the body. Antibodies, another class of proteins, recognize and defend against invaders. Each of these roles is made possible by the diversity of amino acid sequences and the way they fold into shapes uniquely suited to their tasks. It is not an exaggeration to say that proteins are the workhorses of life, carrying out the majority of tasks that allow organisms to grow, adapt, and survive. Carbohydrates, by contrast, are best known as sources of energy. They are composed of simple sugars, or monosaccharides, linked together into longer chains. Glucose, the most familiar of these sugars, circulates in the blood and serves as the primary fuel for cells. When energy is needed quickly, glucose is broken down to release usable power in the form of ATP. Other carbohydrates, such as starch in plants and glycogen in animals, serve as storage forms of energy, ready to be drawn upon when needed. Beyond energy, carbohydrates also provide structure. Cellulose, a carbohydrate made of long chains of glucose, forms the rigid walls of plant cells, giving plants their strength and shape. Chitin, another carbohydrate, forms the exoskeletons of insects and crustaceans. In both energy and structure, carbohydrates remind us that simple repeating units can build powerful and enduring frameworks for life. Lipids form the third great family of macromolecules. Unlike proteins and carbohydrates, lipids are not always made of repeating chains, but are united by their common property. They do not dissolve easily in water. This makes them perfect for forming barriers. The membranes that surround every cell are made of phospholipids, molecules with one end that interacts with water and another end that avoids it. When arranged in layers, they create a stable boundary that protects the inside of the cell from the outside world, while still allowing selective passage of materials. Beyond forming membranes, lipids serve as long-term energy stores. Fats and oils contain dense reserves of energy that can be drawn upon when food is scarce. Lipids also play roles in signaling. Steroids, such as hormones, regulate processes as varied as growth, stress response, and reproduction. In every living organism, lipids provide balance. They store, they protect, and they regulate. The final pillar is nucleic acids, the molecules of information. DNA and RNA, the two types of nucleic acids, carry the instructions for life. They are composed of smaller units called nucleotides, each containing a sugar, a phosphate group, and a nitrogenous base. The sequence of these bases encodes information. Much like letters in a book, DNA stores this information in long, stable chains, preserving it across generations. RNA, often shorter and more versatile, helps translate DNA's instructions into proteins, carrying messages and guiding the assembly of amino acids. Together, DNA and RNA ensure continuity, passing on traits from parent to offspring and guiding the day-to-day functions of cells. Without nucleic acids, life would lack memory, identity, and the ability to evolve. They are the record keepers and storytellers of biology, writing the history of life in chemical code. Although we often study these four families separately, their true power emerges when they work together. In a single cell, carbohydrates provide energy, lipids form the protective boundary, proteins carry out reactions, and nucleic acids store instructions. None of these could sustain life alone, but together they create a dynamic system greater than the sum of its parts. The cooperation among macromolecules illustrates one of the most beautiful aspects of chemistry. Simple units, when combined, give rise to extraordinary complexity. From the smallest bacterium to the largest whale, the same four pillars appear, supporting the full spectrum of life. What is especially remarkable is that all four macromolecules are built from relatively simple ingredients. Proteins are chains of just 20 different amino acids. Carbohydrates are assembled from sugars that share a common formula. Lipids arise from fatty acids and glycerol, small molecules that can combine in predictable ways. Nucleic acids are built from just four bases: adenine, thymine, or uracil, cytosine, and guanine. Out of these modest building blocks, the immense diversity of life is constructed. The pattern is repeated endlessly. Small, simple pieces joining into larger, more complex wholes, each step creating new properties and new possibilities. The story of macromolecules is also the story of cycles. Energy captured by plants in the form of carbohydrates is eaten by animals, converted into proteins and lipids, and then passed back into the environment when life ends. Nucleic acids preserve instructions, which are read and expressed through proteins, which in turn regulate the synthesis of carbohydrates and lipids. These cycles remind us that macromolecules are not static structures but participants in a continuous flow of matter and energy. Life does not pause. It is a ceaseless weaving and unweaving of molecules, guided by the quiet order of chemistry. Within you now, as you lie in stillness, these macromolecules are at work. Proteins are folding into precise shapes, catalyzing reactions that keep your cells balanced. Carbohydrates are being broken down into glucose, fueling your body's quiet rhythm. Lipids form the membranes that keep each cell whole, while also storing reserves of energy for later. Nucleic acids, coiled within the nucleus, are being copied and read, ensuring that every new cell knows its place and purpose. All of this unfolds silently, tirelessly, and without pause. You need not direct it. It happens because macromolecules are reliable, patient, and resilient. To reflect on macromolecules is to see the quiet architecture of life. They are the beams and pillars that hold everything in place. They are not loud or showy, but steady and enduring. They remind us that life's strength comes not from grandeur but from the patient accumulation of simple, reliable structures working in harmony. From the food you eat to the cells that form your body, from the instructions in your DNA to the energy that keeps you alive, the macromolecules are always present, always sustaining, always connecting the pieces of life's great puzzle. And as you drift toward sleep, you may find comfort in knowing that these four pillars are within you, carrying out their quiet roles. They have sustained life for billions of years, across oceans, forests, and generations. They will continue to do so long after this moment, tirelessly maintaining the harmony of existence. Proteins, carbohydrates, lipids, and nucleic acids. The macromolecules stand as the four great pillars of life's chemistry, the silent foundation upon which all living beings rest. 8. Proteins, the body's quiet workers. Within the living world, proteins stand as some of the most remarkable creations of chemistry. They are not only abundant but endlessly varied, and they perform the vast majority of tasks that keep organisms alive. In many ways, proteins are the quiet workers of life, tirelessly building, repairing, transporting, signaling, and protecting, often without recognition. They are found in every cell, in every tissue, and in every living creature, from the simplest bacterium to the most complex human body. To reflect on proteins is to glimpse the delicate machinery that allows life to move, to think, and to endure. At their heart, proteins are chains of smaller molecules known as amino acids. There are 20 standard amino acids used by nearly all living organisms, each one small and relatively simple, yet each carrying its own chemical personality. Some amino acids are hydrophobic, preferring to avoid water, while others are hydrophilic, easily dissolving in it. Some carry positive charges, others negative, and some are neutral. These differences may seem small, but when amino acids are linked together into long chains, the sequence determines how the chain will fold, twist, and coil. That folding creates a unique three-dimensional shape, and the shape determines what the protein can do. The process of building proteins begins with instructions stored in DNA. Each gene provides the code for a specific sequence of amino acids, and through the process of transcription and translation, that code is read and converted into a functioning protein. This flow of information from DNA to RNA to protein is sometimes called the central dogma of molecular biology, and it is one of the most profound insights into the way life operates. The fact that every protein in your body, from those that digest your food to those that allow you to see, is built from the same language of 20 amino acids, is a reminder of both simplicity and unity in biology. Once a protein is formed, its work begins. Some proteins serve as enzymes, catalysts that speed up chemical reactions. Without enzymes, the reactions needed to sustain life would occur too slowly to matter. With enzymes, reactions take place in fractions of a second. For instance, the enzyme amylase, present in saliva, begins the breakdown of starch into sugars as soon as food enters the mouth. Other enzymes in the stomach and intestines continue this process, breaking food down into smaller pieces that can be absorbed and used. At every step, enzymes ensure that reactions happen swiftly and accurately, guiding the flow of metabolism with quiet efficiency. Proteins also play critical structural roles. Collagen, one of the most abundant proteins in the body, provides strength and flexibility to connective tissues, tendons, and skin. Keratin forms hair, nails, and the outer layers of skin, offering protection from the environment. In muscles, proteins, such as actin and myosin, interact to create contraction, allowing movement and strength. Even inside cells, proteins act as scaffolding, forming the cytoskeleton that holds organelles in place and helps cells maintain their shape. Without these structural proteins, the body would lack the strength and form it relies on every moment. Other proteins act as carriers and transporters. Hemoglobin, for example, binds oxygen in the lungs and carries it through the bloodstream to tissues in need. Each hemoglobin molecule can carry 4 oxygen molecules, releasing them where they are required and picking up carbon dioxide in return. Without hemoglobin, oxygen could not be delivered efficiently, and life would falter. In cell membranes, transporter proteins act as gatekeepers, allowing specific molecules to pass in and out, while keeping others at bay. They maintain balance, regulate nutrients, and ensure communication between the cell and its environment. Proteins also serve as messengers in the intricate communication systems of life. Hormones such as insulin, themselves proteins, travel through the blood to signal when glucose levels are high, directing cells to absorb and store the energy. Other protein messengers regulate growth, reproduction, and mood. Receptors, also proteins, sit on the surfaces of cells, waiting for signals to arrive. When a hormone or neurotransmitter binds to a receptor, the protein changes shape, setting off a cascade of events inside the cell. In this way, proteins translate signals from the outside world into actions within, ensuring that the body responds in harmony with its environment. In the immune system, proteins provide defense and protection. Antibodies, specialized proteins produced by white blood cells, recognize and bind to foreign invaders, such as viruses or bacteria. By attaching to these threats, antibodies mark them for destruction and prevent them from causing harm. Other immune proteins act as messengers, calling for reinforcements when infection is present. Still others destroy pathogens directly. This system of defense is vast and intricate, and at every level, proteins are its key players. Even memory and thought depend on proteins. In neurons, proteins form the channels that allow ions to flow, creating the electrical signals that underlie every impulse of the brain. Synaptic proteins guide the release of neurotransmitters, the chemical messengers that allow one neuron to communicate with the next. Over time, changes in proteins at synapses help strengthen or weaken connections, creating the patterns we recognize as learning and memory. From the beating of the heart to the forming of a thought, proteins are present, guiding, and enabling. One of the most striking qualities of proteins is their diversity. A single organism can produce tens of thousands of different proteins, each with its own shape and function. This diversity arises not only from the sequence of amino acids, but also from the way proteins are folded and modified after they are made. Some proteins work alone, while others assemble into complexes with multiple subunits. Together, they create a network of interactions that sustain life at every scale. It is worth noting, too, that proteins are not permanent. They are built, they work, and eventually they are broken down and replaced. This cycle ensures that damaged or outdated proteins do not accumulate, and that the body remains flexible and adaptive. Every day, your body produces and degrades countless proteins, recycling their amino acids into new forms. This quiet turnover is part of life's rhythm, a reminder that stability comes not from stillness, but from constant renewal. As you rest, proteins are at work within you. Enzymes are guiding the reactions that release energy from food. Structural proteins are maintaining the integrity of your tissues. Carrier proteins are moving oxygen, ions, and nutrients to where they are needed. Signaling proteins are regulating balance, ensuring that your internal environment remains stable. Immune proteins are standing guard, ready to protect. Neural proteins are carrying signals that give rise to thoughts, feelings, and dreams. They do all this silently, without pause, and without demand. Proteins are, in essence, the body's quiet workers. They do not shine with the brilliance of stars, nor do they tower like mountains, but their work is no less profound. They embody the principle that strength and beauty arise from precision and patience. In every fold of their shapes, in every bond they form, they carry the quiet wisdom of chemistry. And as you drift deeper into calm, you may find comfort in knowing that these proteins are within you, always working, always supporting, always sustaining the miracle of life. 9. Carbohydrates, the gentle energy sources. Among the macromolecules that sustain life, carbohydrates hold a special place. They are the gentle energy sources of biology, molecules that store the warmth of the sun and release it slowly, steadily, to fuel the countless reactions that keep living organisms alive. Carbohydrates are often simple in structure compared to proteins or nucleic acids, yet their role is no less profound. They provide immediate energy when needed, store reserves for later, and even contribute to the architecture of plants and animals. To reflect on carbohydrates is to understand how life gathers, saves, and spends its energy in quiet cycles that never cease. Carbohydrates are built from simple units called monosaccharides, which are sugars in their most basic form. Glucose is perhaps the most well-known, a 6-carbon sugar that circulates in the blood of animals and serves as the primary fuel for cells. Fructose, found naturally in fruits, though they share the same chemical formula, their atoms are arranged differently, and these subtle differences influence how the body uses them. These simple sugars can join together to form disaccharides, such as sucrose, the familiar table sugar, or lactose, the sugar in milk. With further linking, chains of sugars form polysaccharides, long, complex carbohydrates that serve as energy reserves or structural components. When glucose enters a cell, it does not linger idly. It is quickly put to use in a process called glycolysis, where enzymes break it down into smaller fragments. Each step releases a little energy, which is captured and stored in the form of ATP, the energy currency of life. This ATP then powers muscle contractions, nerve signals, and the synthesis of new molecules. In the presence of oxygen, the breakdown of glucose continues in the mitochondria, releasing even more energy in a controlled flow. It is through this process that the energy captured by plants in photosynthesis becomes available to animals, fungi, and countless microorganisms. A single molecule of glucose may seem small, but within it lies the potential to sustain life. Carbohydrates also serve as storage forms of energy, waiting patiently until they are needed. In plants, glucose molecules are linked into long chains of starch, coiled and packed into grains within roots and seeds. In animals, glucose is stored as glycogen, a similar polysaccharide that is kept mainly in the liver and muscles. When energy is required, enzymes quickly break down glycogen, releasing glucose into the bloodstream or directly into active tissues. This ability to store and release energy ensures that organisms can endure times when food is scarce or when sudden bursts of activity demand more fuel. In this way, carbohydrates act not only as immediate providers but also as quiet guardians of energy balance. Beyond their role in energy, carbohydrates contribute to structure. In plants, cellulose forms the rigid walls that surround each cell, giving leaves their shape and stems their strength. Cellulose is made of long chains of glucose, linked in a way that resists digestion by most animals. This resistance provides durability, allowing trees to stand tall and grasses to bend without breaking. In insects and crustaceans, another carbohydrate called chitin forms protective exoskeletons. Like cellulose, chitin is built from repeating sugar units, but its structure has been adapted for strength and resilience. These examples remind us that carbohydrates are not only fuels but also the materials from which life builds its shelter. Carbohydrates also play roles in recognition and communication. On the surfaces of cells, small chains of sugars are attached to proteins and lipids, forming structures that act as identifiers. These carbohydrate markers allow cells to recognize one another, distinguishing self from non-self, and guiding interactions between tissues. The immune system, for example, relies on these sugar patterns to detect invaders and respond appropriately. Even in the nervous system, carbohydrates participate in cell signaling, helping guide the growth and connection of neurons. Though often overlooked, these sugar decorations are vital for the orchestration of complex biological systems. What makes carbohydrates especially gentle is their availability. Plants produce them in abundance through photosynthesis, capturing sunlight and transforming carbon dioxide and water into sugars. These sugars become fruits, grains, and roots, nourishing not only the plants themselves but also the animals and humans who consume them. The cycle continues when animals eat plants, breaking down the sugars and using them for energy, then releasing carbon dioxide back into the atmosphere. That carbon dioxide becomes the starting point for photosynthesis once again, completing the cycle. Carbohydrates, in this way, serve as the quiet mediators between the energy of the sun and the energy of life. It is worth considering the diversity of carbohydrates and how each form contributes in its own way. Simple sugars such as glucose and fructose provide quick bursts of energy, easily absorbed and rapidly metabolized. Disaccharides like sucrose and lactose offer slightly longer chains, providing a steady source of sweetness and fuel. Polysaccharides such as starch and glycogen act as reserves, waiting patiently for moments of need. Structural carbohydrates like cellulose and chitin offer strength and durability, protecting and supporting organisms in their growth. Each variation shows how a simple unit, a sugar molecule, can be adapted to serve multiple roles, from nourishment to defense. Carbohydrates also remind us of the balance between abundance and moderation. Because they are efficient energy sources, organisms have evolved to store and use them carefully. In humans, blood glucose levels are maintained within narrow limits, regulated by hormones like insulin and glucagon. Too little glucose, and cells are deprived of fuel. Too much, and delicate tissues may be harmed. This balance reflects the precision of life's chemistry, where even the simplest molecules require careful control to maintain harmony. As you rest, carbohydrates are quietly at work within you. Glucose molecules flow through your bloodstream, entering cells to be broken down into energy. Glycogen stores in your liver and muscles remain ready, prepared to release glucose if the body calls for it. Cellulose in the food you have eaten passes through your digestive system, providing fiber that supports balance and health. Carbohydrate markers on the surfaces of your cells silently guide interactions, ensuring that your immune system recognizes friend from foe. Every moment, these molecules are present, gentle, and reliable, sustaining you without need for attention. To reflect on carbohydrates is to recognize the softness of life's chemistry. They are not dramatic or rare. They are simple, abundant, and essential. They provide energy for movement, thought, and growth. They build the walls of plants, the shells of animals, and the patterns of recognition between cells. They connect the energy of the sun to the energy of life, forming one of the most important bridges in the natural world. Carbohydrates are truly the gentle energy sources, the quiet companions of every living being. As you drift deeper into calm, you may find peace in knowing that carbohydrates, in all their forms, are within you and around you, steady and dependable. They have supported life for billions of years, flowing through cycles of growth, decay, and renewal. And they continue their work even now, sustaining the quiet balance of your body, ensuring that life moves forward in harmony. 10. Lipids, barriers, and energy reserves. Among the great macromolecules of life, lipids occupy a place that is both quiet and essential. They are not as obviously active as proteins, nor as information-rich as nucleic acids, nor as sweetly immediate as carbohydrates. Yet lipids serve as barriers, boundaries, and reserves. They store energy for times when it is needed most. They form the membranes that separate the inside of a cell from the outside world. They insulate, protect, and regulate. Lipids are the silent guardians of stability, ensuring that life remains balanced and protected. To consider lipids is to see how chemistry provides both shelter and security, how simple molecules can give rise to both structure and sustenance. Lipids are defined less by a single structure and more by a shared property. They do not dissolve easily in water. This characteristic, known as hydrophobicity, arises because lipids are composed mainly of carbon and hydrogen atoms, linked together in long chains or compact rings. These atoms share electrons evenly, leaving little polarity, and thus they do not interact well with the polar molecules of water. Instead, lipids prefer to associate with one another, forming droplets, layers, or membranes. It is this tendency that allows lipids to act as barriers, creating boundaries that enclose life's chemistry within cells. The most familiar form of lipids are fats and oils, known more precisely as triglycerides. Each triglyceride consists of a glycerol molecule attached to 3 fatty acids. Fatty acids themselves are long chains of carbon and hydrogen, with a small acidic group at one end. When 3 fatty acids attach to glycerol, the result is a compact, energy-rich molecule. Fats store more than twice the energy per gram compared to carbohydrates or proteins, making them efficient, long-term reserves. In animals, fat is stored in adipose tissue, where it provides energy during times of scarcity. It also cushions organs, protecting them from shock, and insulates the body, helping to maintain stable temperatures. Oils in plants serve a similar purpose, stored in seeds to provide nourishment for germination and growth. The structure of fatty acids also determines whether a fat is solid or liquid at room temperature. Saturated fatty acids, with no double bonds between carbon atoms, form straight chains that pack closely together, creating solid fats, such as butter. Unsaturated fatty acids, with one or more double bonds, bend at those points, preventing tight packing and creating liquids like olive oil. These differences not only influence texture and appearance, but also affect how fats interact within living systems. The flexibility of unsaturated fats, for example, helps maintain fluidity in cell membranes, ensuring that they remain dynamic rather than rigid. Beyond triglycerides, another important class of lipids is phospholipids. These molecules resemble fats, but with a key difference. One of the fatty acid chains is replaced by a phosphate group, which carries a negative charge. This creates a molecule with two distinct ends, one hydrophobic and one hydrophilic. When placed in water, phospholipids spontaneously arrange themselves into bilayers, with hydrophobic tails facing inward and hydrophilic heads facing outward. These bilayers form the membranes of cells, delicate yet resilient barriers that separate the inner chemistry of life from the external environment. Within these membranes, proteins are embedded, channels are formed, and signals are received. But it is the lipids themselves that create the foundational structure. Without phospholipids, cells could not exist. Another category of lipids includes steroids, molecules built not from long chains but from 4 interconnected rings. Cholesterol is the most familiar example. Though often spoken of in connection with health concerns, cholesterol plays vital roles in biology. It is an essential component of cell membranes, helping to regulate their fluidity and stability. Cholesterol is also the starting point for the synthesis of steroid hormones, such as estrogen, testosterone, and cortisol. These hormones, though small in amount, have powerful effects, regulating reproduction, metabolism, stress responses, and more. All of these functions rest upon the quiet chemistry of lipids, whose structures allow them to influence systems far larger than themselves. Lipids also play roles in signaling beyond hormones. Certain lipids act as messengers within cells, transmitting information from the membrane to the interior. These signaling lipids help regulate growth, immune responses, and even programmed cell death. They act in small bursts, appearing and disappearing as needed, guiding processes that keep cells balanced. Other lipids form protective coatings. Waxes, for instance, are lipids that provide waterproof barriers. On the leaves of plants, waxes prevent excessive water loss. In animals, waxes line fur, feathers, and skin, offering protection against the elements. Though often unnoticed, these coatings are essential for survival in challenging environments. The quiet power of lipids can also be seen in their role as reservoirs of energy. Unlike carbohydrates, which provide quick bursts of fuel, lipids release energy slowly and abundantly. During fasting or extended activity, stored fats are broken down into fatty acids and glycerol, which are then used by cells to generate ATP. This process allows organisms to endure when immediate food is unavailable. Migrating birds, for example, rely heavily on fat reserves to sustain them on long flights. Hibernating animals live off stored fat for months, using it not only for energy, but also for warmth. Even in humans, the fat stored in adipose tissue is a silent safeguard, ensuring that the body has reserves to draw upon in times of need. Lipids also remind us of balance. Too little fat, and organisms struggle to store energy, regulate temperature, or absorb fat-soluble vitamins, such as A, D, E, and K. Too much fat, stored excessively, can burden the body and disrupt systems. Life thrives in the middle, in the steady presence of lipids that provide enough, but not too much. This balance mirrors the chemistry of life itself, where equilibrium is the key to survival. What is most striking about lipids is how they blend structure and function. They are at once barriers and stores, protectors and providers. They create boundaries that define cells, yet they also serve as reserves that sustain organisms through hardship. They act as messengers, guiding growth and responses, and as insulators, keeping life warm and safe. Their diversity arises from variations in simple building blocks, fatty acids, glycerol, phosphate groups, and rings combined in different ways to serve different needs. Out of this simplicity comes versatility, a hallmark of life's chemistry. As you rest, lipids are at work within you. Membranes made of phospholipids protect every cell, holding in the fluids and molecules that sustain life. Cholesterol stabilizes those membranes, ensuring they remain fluid but not fragile. Hormones derived from lipids travel through your blood, quietly regulating countless processes. Fat stored in tissues waits patiently, ready to provide energy if it is needed. Even the oils in your skin act as barriers, preventing water loss and shielding you from the outside world. Every moment, lipids are present, silent, but essential, carrying out their roles without demand for recognition. To think of lipids is to think of the quiet guardians of life. They are not dramatic, yet their influence is profound. They form the boundaries that define living beings, the reserves that sustain them, and the signals that guide their growth and balance. They embody the principle that stability and flexibility can coexist, that strength can be soft, and that protection can be silent. As you drift into calm, you may imagine the lipids within you, forming gentle layers around your cells, storing warmth and energy, guiding signals, and keeping balance. They are there always, as they have been for countless generations, carrying out their tasks with patience and constancy. Lipids are barriers and reserves, protectors and providers, the quiet chemistry of life at its most steadfast. 11. Nucleic acids, the story of life. Written in code, among the macromolecules that form the foundation of life, nucleic acids are the storytellers. They hold the instructions for building every cell, every protein, and every living organism. They preserve memory across generations, ensuring that traits are passed from parent to offspring, that continuity persists even as time moves forward. DNA and RNA, the two great forms of nucleic acids, are not large in variety compared to proteins or carbohydrates, but their significance is immeasurable. They are the scriptwriters of existence, the carriers of life's code, the gentle threads that weave the story of biology from the dawn of life to this very moment. Nucleic acids are polymers, chains made from smaller units called nucleotides. Each nucleotide is composed of three parts: a sugar, a phosphate group, and a nitrogenous base. The sugar and phosphate form the backbone of the chain, while the base extends outward like a letter in a sentence. There are four main bases in DNA: adenine, thymine, cytosine, and guanine. In RNA, thymine is replaced by uracil. Though these bases are simple, their sequences encode the vast diversity of life. The arrangement of bases in a strand of DNA is like the letters of an alphabet, spelling out instructions that cells can read and carry out with precision. The structure of DNA is one of the most elegant discoveries in science. It takes the form of a double helix, two strands coiled around each other, with bases paired in the center. Adenine pairs with thymine, guanine pairs with cytosine, each bond stabilized by delicate hydrogen interactions. This pairing ensures that the code can be copied with fidelity, as each strand serves as a template for the other. The helical shape, compact and graceful, allows enormous amounts of information to be stored in a remarkably small space. Within the nucleus of a single human cell, 2 meters of DNA are coiled and folded into chromosomes, all fitting within a space invisible to the naked eye. Yet, within this compact form lies the full set of instructions for building an entire organism. RNA, though similar in structure, serves different roles. It is often shorter, single-stranded, and more flexible than DNA. Messenger RNA carries information from DNA to the ribosomes, the molecular machines that assemble proteins. Transfer RNA brings the correct amino acids into place during protein synthesis, ensuring that the sequence follows the code precisely. Ribosomal RNA, part of the ribosome itself, helps catalyze the formation of bonds between amino acids. Together, these forms of RNA act as interpreters, taking the instructions written in DNA and turning them into functional proteins. RNA, in this way, is the bridge between information and action, between script and performance. The significance of nucleic acids extends beyond their chemistry. They embody continuity. Every living organism on Earth carries DNA that connects it to ancestors, stretching back billions of years. The same genetic code that shapes humans also shapes plants, animals, fungi, and bacteria. The universality of the code speaks to a shared origin, a common ancestor from which all life descends. Differences in sequences give rise to diversity, but the underlying system remains the same. In this sense, DNA is a unifying thread, binding all living things into one vast and ancient story. The process of replication ensures that this story is preserved. When a cell divides, its DNA must be copied so that each daughter cell receives a complete set of instructions. Enzymes unwind the double helix, separate the strands, and use them as templates to build new complementary strands. The result is two identical molecules of DNA, each ready to guide the life of a new cell. Though errors can occur, the system is remarkably accurate, and even mistakes can contribute to evolution by introducing variation. Thus, replication is not only a mechanism of continuity but also a source of change, allowing life to adapt and evolve over time. Nucleic acids are also deeply involved in regulation. Segments of DNA called genes provide instructions, but other regions act as switches, turning genes on or off depending on the needs of the cell. Proteins called transcription factors bind to these regions, guiding whether RNA is produced. In this way, the code is not static, but dynamic, responding to signals, adapting to conditions, and ensuring balance. A skin cell and a nerve cell carry the same DNA, yet they express different genes, creating different forms and functions. This selective reading of the code allows a single genetic library to produce the immense variety of cell types in a multicellular organism. Beyond individual organisms, nucleic acids shape the flow of life on Earth. Mutations, small changes in DNA sequences, accumulate over time. Some are harmful, others neutral, and a few beneficial. Those beneficial changes, preserved and passed on, drive evolution, allowing species to adapt to their environments. Through this process, the story written in DNA is not fixed but ever evolving, adding new chapters across the ages. From the first simple cells to the complexity of humans, nucleic acids have carried the narrative forward, preserving what is essential while allowing room for change. The quiet persistence of nucleic acids can also be seen in their stability. DNA, protected within the nucleus, can endure for years, even centuries, under the right conditions. Fragments of DNA recovered from ancient bones have revealed the genetic makeup of long-extinct species, showing that the story of life is written not only in the living, but also in the remnants of the past. RNA, though less stable, offers flexibility, able to appear and disappear as needed, carrying messages and catalyzing reactions with agility. Together, DNA and RNA provide both permanence and adaptability, the balance that life requires. As you rest, nucleic acids are active within you. DNA in your cells is coiled into chromosomes, quietly preserving the instructions that guide your growth, repair, and function. Segments of this DNA are being read into RNA, which moves outward to direct the making of proteins. In ribosomes, amino acids are being linked together according to these instructions, creating enzymes, hormones, and structural components. All of this unfolds silently, without your awareness, and yet it sustains you every moment. To reflect on nucleic acids is to recognize the elegance of life's design. A small set of molecules, repeating in endless combinations, carries the vast instructions for every living being. They are both ancient and current, both stable and adaptable. They are the archive of the past and the blueprint of the future. Within their sequences lies the history of life on Earth, the diversity of species, and the potential for generations yet to come. As you drift into calm, you may take comfort in knowing that within every cell of your body lies this quiet code, written in a simple alphabet of 4 bases, endlessly copied, endlessly expressed, endlessly renewed. It is the story of life written in chemistry, the gentle thread that ties you to every living thing, past and present. DNA and RNA, the nucleic acids, carry this story forward, silently and faithfully, sustaining the miracle of existence. 12. Enzymes, the quiet catalysts. In the vast and intricate chemistry of life, there are countless reactions taking place every second. Nutrients are broken down into smaller pieces, energy is captured and stored, signals are transmitted, molecules are built up, and wastes are dismantled. Left to occur on their own, many of these reactions would proceed too slowly to sustain life. Some might take years, or even centuries, to happen spontaneously. Yet within every living organism, they occur in fractions of a second, in perfect sequence, and in harmony with the needs of the moment. This extraordinary acceleration is made possible by enzymes. They are proteins, but of a very special kind. They are catalysts, substances that speed up reactions without being consumed themselves. In this way, enzymes are the quiet facilitators of life, ensuring that chemistry moves at just the right pace. At their heart, enzymes work by lowering the activation energy of reactions. Every chemical change requires a small push, an initial investment of energy to break existing bonds and allow new ones to form. Without this push, molecules may remain as they are, unwilling to rearrange. Enzymes provide the conditions for this push to happen more easily, guiding molecules into positions where bonds can be broken and reformed with less effort. In doing so, enzymes transform reactions that would be impossibly slow into processes that unfold rapidly enough to keep organisms alive. The structure of an enzyme is central to its function. Like all proteins, enzymes are made of chains of amino acids folded into precise 3-dimensional shapes. Within this shape is a special region called the active site. It is here that the reacting molecules, known as substrates, bind. The active site is often compared to a lock and the substrate to a key. Only molecules with the right shape and chemical properties can fit and be acted upon. When the substrate enters the active site, the enzyme may change shape slightly, a process called induced fit, tightening around the substrate to guide the reaction more effectively. Once the reaction has occurred, the products are released, and the enzyme is free to act again. This cycle can repeat thousands or even millions of times, making enzymes extraordinarily efficient. Consider the enzyme catalase, found in nearly all living cells. Its role is to break down hydrogen peroxide, a byproduct of metabolism that can be harmful if allowed to accumulate. Without catalase, hydrogen peroxide would linger and damage cells. With catalase, the reaction occurs almost instantly, producing harmless water and oxygen. A single catalase molecule can act on millions of hydrogen peroxide molecules every second. This efficiency is not unique to catalase. Many enzymes are capable of extraordinary feats, turning what might seem like improbable reactions into routine steps of daily life. Enzymes are involved in every aspect of metabolism. In digestion, enzymes break down large molecules of food into smaller components that can be absorbed. Amylases split starch into sugars. Proteases dismantle proteins into amino acids, and lipases separate fats into glycerol and fatty acids. These smaller molecules can then be used to build new structures or burned for energy. Without digestive enzymes, the food we eat would remain locked in complex molecules, unavailable for the body's needs. Enzymes unlock that potential, releasing the nourishment hidden within. In the synthesis of molecules, enzymes play an equally vital role. To build DNA, for example, enzymes link nucleotides together in precise sequences, ensuring that the genetic code is faithfully copied. To produce proteins, enzymes help assemble amino acids in the right order, forming bonds that create long chains. To generate ATP, enzymes guide reactions that transfer energy from one molecule to another. In each case, enzymes are the directors of chemistry, guiding molecules into place, stabilizing transitions, and ensuring accuracy. Enzymes also allow for regulation, because they can be turned on or off, sped up or slowed down. Enzymes provide control over when and how reactions occur. Some enzymes are activated only in the presence of certain molecules, acting like switches that respond to the cell's needs. Others are inhibited when enough product has been made, preventing waste and maintaining balance. This regulation ensures that metabolism does not run chaotically, but proceeds with order and efficiency. Cells conserve resources, respond to changing conditions, and maintain homeostasis through the careful management of enzymes. The specificity of enzymes is another remarkable quality. Each enzyme typically acts on only one substrate or on a small group of closely related molecules. This precision ensures that reactions occur in the right place at the right time, without unintended consequences. Imagine a vast city with countless activities happening simultaneously. If workers could not distinguish between tasks, the city would fall into confusion. But with specialization, each worker knows their role, and the city thrives. In the same way, enzymes provide specialization within the chemistry of life, each one dedicated to a particular reaction, each one ensuring that the great city of the cell operates smoothly. Yet enzymes are not rigid. They are flexible, able to adapt to new conditions, and even evolve new functions over time. Mutations in DNA may alter the sequence of amino acids, changing the shape of an enzyme's active site. Sometimes this change is harmful, but at other times it creates new abilities, allowing organisms to adapt to new environments. In this way, enzymes are not only facilitators of life, but also participants in evolution. They reflect both stability and change, continuity and adaptability. The importance of enzymes can also be seen in how their absence or malfunction affects life. Many genetic disorders are the result of missing or defective enzymes. Where a single step in metabolism is disrupted, these conditions highlight how dependent we are on the quiet work of enzymes, how even one missing catalyst can create imbalance. They also remind us of the delicate precision of life's chemistry, where every reaction has its place and every enzyme its role. As you rest, enzymes are active within you. They are breaking down glucose to release energy, building new molecules to repair tissues, copying DNA to prepare for cell division, and dismantling wastes to keep balance. They work in every cell, in every organ, tirelessly and silently. You do not feel them, yet they sustain you in every moment. Their work is gentle but constant, ensuring that the chemistry of life proceeds smoothly as you drift deeper into stillness. To think of enzymes is to think of the quiet catalysts of existence. They are not consumed by their work, yet they carry it out endlessly. They lower barriers, making the improbable possible, and they do so with precision and grace. They are patient, repeating their tasks millions of times without error. They remind us that life is not only built on structure but also on process, on the flow of reactions guided by unseen hands. As you sink into calm, you may imagine these catalysts within you, gently guiding molecules into place, lowering the barriers, and smoothing the path for reactions to unfold. They are the silent facilitators of your life, the quiet workers of your chemistry, the patient companions of your cells. Enzymes are the quiet catalysts, the unsung yet indispensable forces that make life not only possible, but beautifully efficient. 13. Energy in biochemistry. ATP, the gentle currency. Every living system requires energy. It is the quiet current that flows through every cell, powering growth, repair, thought, movement, and rest. Without energy, even the simplest reaction would halt, and life would dissolve into stillness. Yet energy must be managed carefully. It cannot be allowed to rush uncontrolled, nor can it be stored too loosely. Cells require a way to capture energy, to store it in small, usable amounts, and to release it precisely where and when it is needed. This role is filled by a molecule called adenosine triphosphate, or ATP. It is not large or glamorous, but it is indispensable. ATP is often called the energy currency of life, for it provides the medium of exchange that allows energy to flow smoothly through biology. ATP is a nucleotide, a molecule built from 3 parts: a sugar called ribose, a nitrogenous base called adenine. The first 2 components, ribose and adenine, form adenosine, the stable foundation. Attached to this base are the phosphates, each carrying negative charges that repel one another. Holding these phosphates together requires energy, and the bonds between them are often described as high-energy bonds. When one of these bonds is broken, the stored energy is released, and ATP is converted into ADP, adenosine diphosphate, with 2 phosphates. If another bond is broken, ADP becomes AMP, adenosine monophosphate. These transformations provide small, manageable bursts of energy that cells can use directly. The beauty of ATP lies in its universality. Nearly every process in every living organism relies on ATP as its source of energy. When a muscle contracts, ATP is hydrolyzed to provide the power for actin and myosin to slide past each other. When a neuron fires, ATP fuels the pumps that restore ion balances across membranes. When DNA is copied, when proteins are synthesized, when molecules are transported across membranes, all of these processes draw upon ATP. It is the common currency that allows cells to pay the energetic cost of their activities. Without it, The economy of life would collapse. ATP is not stored in large amounts but is regenerated continuously. Each molecule of ATP is used and replaced in seconds. In humans, the total amount of ATP present in the body at any moment is relatively small, yet the turnover is immense. The average person recycles their body weight in ATP every day, as molecules are used, converted to ADP, and then rebuilt again from nutrients. This rapid cycle ensures that energy is always available but never wasted, always circulating like a gentle stream through the pathways of metabolism. The regeneration of ATP comes primarily from cellular respiration. In this process, glucose and other fuels are broken down in stages, and the energy released is used to rebuild ATP from ADP and phosphate. The first stage, glycolysis, splits glucose into smaller fragments, releasing a modest amount of ATP. In the presence of oxygen, these fragments enter the mitochondria, where the citric acid cycle and oxidative phosphorylation generate much larger amounts. Electrons are passed along chains of proteins, and the energy they release is harnessed to pump protons across a membrane. As the protons flow back, ATP synthase, a remarkable molecular machine, captures the energy to join phosphate to ADP, forming new ATP. This process is efficient and constant, ensuring a steady supply of energy for the cell's needs. In the absence of oxygen, cells can still produce ATP through fermentation, though less efficiently. Muscle cells, for instance, switch to fermentation during intense exercise when oxygen is scarce, producing ATP quickly but in limited amounts. Other organisms, such as certain bacteria and yeasts, rely on fermentation as their primary means of ATP production. In every case, the goal remains the same: to keep the energy currency flowing to ensure that ATP is available when required. ATP's importance extends beyond energy transfer. It also serves as a signaling molecule. Inside cells, the concentration of ATP reflects the energy state, and enzymes respond accordingly, turning pathways on or off depending on availability. ATP can also act as a substrate, donating phosphates to proteins in a process called phosphorylation. This modification can change the shape and activity of proteins, allowing cells to regulate processes with precision. Through these roles, ATP becomes not only a carrier of energy, but also a messenger of balance, guiding the flow of metabolism and communication. The elegance of ATP lies in its simplicity. It is small, easily made, and easily broken. The energy it provides is neither too large nor too small, but just right for the needs of cellular reactions. It can be moved quickly from one part of the cell to another, delivered to enzymes, motors, and pumps as needed. Its universality allows all parts of metabolism to connect, each process drawing from the same pool, each contributing to the cycle of renewal. ATP is not wasteful, nor excessive. It is steady, reliable, and perfectly suited to its role. ATP also illustrates the interconnectedness of life. Plants capture sunlight and use it to produce glucose, which animals then consume. The glucose is broken down to regenerate ATP, fueling movement, thought, and growth. The carbon dioxide and water released return to the environment, to be taken up by plants again. In this cycle, ATP is the intermediary. In the capture of energy and its use, it is a bridge between the environment and the organism, between the sun's radiance and the beating of a heart. As you rest, ATP is at work within you. Molecules of glucose are being dismantled, their energy stored in ATP. That ATP is fueling the contraction of your diaphragm as you breathe, the firing of neurons as you dream, the synthesis of proteins that repair and renew your tissues. Each second, Trillions of ATP molecules are being used and replaced, flowing through you like a constant tide. You do not feel it, yet it sustains every moment of your existence. To reflect on ATP is to appreciate the quiet economy of life. It is a molecule that spends itself endlessly, only to be renewed again. It is the gentle coin of energy, exchanged in every cell, in every reaction. It represents balance, for it holds energy securely, yet releases it easily when needed. It represents unity, for it is shared across all organisms, from the smallest bacterium to the largest whale. And it represents constancy, for it has sustained life for billions of years, through cycles of consumption and renewal. As you drift deeper into calm, you may imagine ATP as a soft current flowing through you, carrying energy where it is needed, never ceasing, never failing. It is the gentle currency of life, the molecule that allows chemistry to become biology, that allows matter to become thought, movement, and being. In every heartbeat, in every breath, in every quiet moment, ATP is there, patiently sustaining the miracle of life. 14. The flow of metabolism. Life is movement, but not always in ways that can be seen. Beneath the stillness of rest, beneath the calm rhythm of breathing, an entire world of motion unfolds within each cell. Molecules are built and broken, energy is stored and released, waste is cleared away, and balance is preserved. This ceaseless motion is called metabolism, the sum of all chemical processes that sustain life. Metabolism is not a single pathway or reaction, but an intricate flow, weaving through countless steps, each one guided by enzymes, each one interconnected with others. It is the river of chemistry that flows through every living being, silent and tireless, ensuring that existence continues moment by moment. At its heart, metabolism is about transformation. Nutrients from food or the environment are taken in, and through reactions they are converted into energy and building blocks. These transformations can be divided into two broad categories: catabolism and anabolism. Catabolism refers to the breaking down of molecules, releasing energy stored in chemical bonds. Anabolism refers to the building up of molecules, using energy to create new structures. Together, these two halves of metabolism form a cycle, one dismantling and releasing, the other constructing and investing, both working in harmony to sustain life. Catabolism begins when energy-rich molecules, such as carbohydrates, fats, or proteins, are broken down. In glycolysis, glucose is split into smaller fragments, releasing energy and producing intermediates that feed into further reactions. In the citric acid cycle, these fragments are processed further, generating electrons that carry energy into the electron transport chain. There, in the mitochondria, the flow of electrons is used to create ATP, the currency of energy. Catabolism is like the gentle dismantling of structures, each step yielding usable energy that cells can spend on their needs. It is a controlled, precise process, ensuring that energy is released gradually, never in wasteful bursts. Anabolism, on the other hand, is constructive. It takes small molecules and links them together into larger, more complex forms. From amino acids come proteins, from nucleotides come nucleic acids, from sugars come polysaccharides, and from fatty acids and glycerol come lipids. Each step requires energy, often provided by ATP, and each step builds the architecture of life. Anabolism is how cells grow, repair themselves, and adapt. When you heal a wound, when you form new memories, when plants stretch toward the sun, it is anabolism at work, quietly constructing from the raw materials of chemistry. The balance between catabolism and anabolism is crucial. Too much breaking down without building up, and life wastes away. Too much building without sufficient energy, and processes falter. The harmony of metabolism ensures that both occur in proportion, guided by signals and needs. Hormones like insulin and glucagon help regulate this balance in humans, directing cells to store energy after a meal or release it during fasting. Other signals within cells fine-tune the flow, turning pathways on or off depending on the availability of nutrients and the demands of the moment. Metabolism is not only about energy but also about pathways that interconnect in networks. The glucose broken down in glycolysis may provide energy, but it may also provide intermediates for synthesizing amino acids. Fatty acids can be oxidized for energy, or they can be used to build membranes. Proteins can be dismantled for fuel, or their components can be recycled into new proteins. Nothing is wasted. The flow of metabolism is circular, with products of one pathway becoming substrates for another, like tributaries feeding into rivers, and rivers feeding back into the sea. This interconnection gives life resilience, allowing it to adapt to changing conditions and make use of whatever resources are available. Another remarkable feature of metabolism is its efficiency. Reactions are organized into sequences where the product of one step becomes the starting point of the next. This prevents the accumulation of useless intermediates and ensures that energy is captured and used effectively. Enzymes guide each step, lowering barriers and maintaining precision. In mitochondria, for example, the electron transport chain couples the flow of electrons to the pumping of protons, and this gradient of protons is then used by ATP synthase to produce ATP. Every step is coordinated, every energy transfer harnessed. It is a masterpiece of quiet engineering, honed over billions of years of evolution. Metabolism also reflects cycles that extend beyond the individual cell. The carbon you exhale as carbon dioxide becomes the carbon that plants take in for photosynthesis. The oxygen plants release returns to animals and humans, fueling respiration. Nitrogen cycles through soil, bacteria, plants, and animals, transformed at each stage by metabolic reactions. These cycles remind us that metabolism is not isolated within organisms but is part of a larger web, connecting all life to the planet and even to the atmosphere. The flow of metabolism is both personal and universal, linking each breath and heartbeat to the wider balance of Earth. During rest, metabolism does not stop. It shifts, conserving energy, repairing tissues, consolidating memories, and renewing molecules. As you sleep, anabolic processes increase, building proteins and restoring balance. Cells quietly recycle damaged components, replacing old molecules with new. Energy is still required, and catabolic processes continue, but the emphasis changes, reflecting the body's needs at that time. Metabolism is adaptive, responding not only to food and activity, but also to the rhythms of day and night. It is attuned to cycles larger than itself, adjusting to ensure harmony. Metabolism also reveals the unity of life. The pathways of glycolysis, the citric acid cycle, and oxidative phosphorylation are nearly universal, found in organisms as different as bacteria and humans. This universality speaks to a common origin, a shared ancestral chemistry that has endured for billions of years. Despite the diversity of life, the core of metabolism remains the same, testifying to its effectiveness and necessity. It is a reminder that all living things are connected not only through the environment but also through the chemistry that flows within them. As you rest now, metabolism is flowing within you. Molecules from food you have eaten are being broken down, their energy captured in ATP. Proteins are being rebuilt from amino acids, lipids reshaped into membranes, DNA copied and repaired. Waste is being dismantled and cleared, balance maintained. It all happens silently, without conscious effort. Yet it sustains your body, your thoughts, and your dreams. You are a river of chemistry, always moving, always renewing, even in stillness. To reflect on metabolism is to recognize the flow of life itself. It is a gentle current, sometimes building, sometimes breaking, but always moving forward. It is balance, efficiency, and resilience, woven into every living being. It connects the smallest cell to the largest ecosystem, the moment of breath to the cycle of the planet, and as you drift deeper into calm, you may find peace in knowing that this flow continues within you, steady and quiet, carrying you through the night and into the day beyond. 15. pH and balance in the body. Life depends on balance. It is not only the balance of energy in and out, or the balance between growth and repair, but also the balance of chemistry itself. Among the most delicate of these balances is the control of pH, the measure of how acidic or basic a solution island. Within living systems, even the smallest shifts in pH can alter reactions, disrupt proteins, or endanger survival. Yet despite this sensitivity, organisms maintain their internal pH with remarkable steadiness. It is one of the quiet triumphs of biology, the ability to keep chemistry in harmony, even as the environment shifts and challenges arise. pH is a scale that measures the concentration of hydrogen ions in a solution. It ranges from 0 to 14, with lower numbers representing higher acidity, higher numbers representing higher basicity, and 7 representing neutrality. Pure water, for instance, has a pH close to 7, perfectly balanced between acids and bases. Acids are substances that donate hydrogen ions, while bases are those that accept them. In the chemistry of life, both acids and bases have roles, but they must be kept in proportion. Too much of either, and delicate systems begin to falter. In the human body, different fluids and compartments maintain characteristic pH levels. Blood, for instance, is kept within a narrow range of about 7.35 to 7.45. Slightly basic, but very close to neutral. This narrow window is crucial. If blood becomes even slightly more acidic or basic, proteins may lose their shapes, enzymes may cease to function properly, and cells may struggle to survive. The stomach, by contrast, maintains a highly acidic environment with pH values around 2, necessary for breaking down food and defending against harmful microbes. In the small intestine, the environment shifts back to near neutral, allowing enzymes to work effectively in digestion. Inside lysosomes, the small compartments that recycle cellular materials, acidity is maintained to help break down waste. Each space has its own balance, tuned precisely to its purpose and maintained with constant care. This control of pH is achieved through buffering systems, processes that resist changes in acidity or basicity. The most important buffer in the human body is the bicarbonate system. When hydrogen ions increase, threatening to make blood more acidic, bicarbonate binds to them, forming carbonic acid, which can then be converted to carbon dioxide in water. When hydrogen ions decrease, making blood more basic, carbonic acid releases hydrogen ions to restore balance. This reversible dance keeps blood pH within its narrow range, moment by moment, breath by breath. In fact, the act of breathing is closely tied to pH. Exhaling carbon dioxide removes acid from the body, while holding the breath allows it to accumulate. In this way, respiration itself becomes a quiet regulator of balance. Proteins also contribute to buffering. Many amino acids have side groups that can accept or donate hydrogen ions, helping to stabilize pH in the fluids where they reside. Hemoglobin, the protein that carries oxygen in red blood cells, is an especially important buffer. It not only binds oxygen but also helps carry carbon dioxide and stabilize the acidity of blood. Phosphate buffers, present in cells and in the kidneys, provide another layer of protection, ensuring that even local changes in pH are corrected quickly. The kidneys themselves play a central role in long-term pH regulation. By excreting hydrogen ions and reabsorbing bicarbonate, they adjust the balance according to the body's needs. This process is slower than breathing but provides a powerful way to maintain stability over hours and days. Together, the lungs and kidneys form a partnership, one adjusting pH rapidly, the other providing steady correction over time. This cooperation illustrates the layered defenses that protect the chemistry of life from disruption. The importance of pH balance can be seen in what happens when it is disturbed. If blood becomes too acidic, a condition known as acidosis, systems begin to fail. Enzymes lose efficiency, nerves struggle to transmit signals, and muscles weaken. If blood becomes too basic, called alkalosis, the effects are equally disruptive, leading to overexcited nerves, muscle spasms, and confusion. Both conditions are dangerous, yet the body's buffering systems usually prevent them, maintaining the narrow range in which life thrives. This resilience is a testament to the quiet precision of biology. Beyond the human body, pH plays a vital role in ecosystems. Lakes and rivers must maintain certain ranges for fish and plants to survive. Soil pH determines which nutrients are available to plants, shaping forests and fields. Oceans, too, depend on stable pH, yet they are sensitive to changes from carbon dioxide dissolving into the water, forming carbonic acid. These shifts ripple through marine ecosystems, affecting corals, shellfish, and countless other forms of life. Just as within the body, balance is essential, and just as within the body, buffers and cycles strive to maintain harmony. On the molecular scale, the significance of pH becomes even more evident. Proteins fold into their functional shapes because of interactions between charged groups, interactions that depend on pH. If acidity changes, these interactions may be altered, and the protein may lose its structure, a process called denaturation. Enzymes, too, have optimal pH ranges in which they function best. The stomach enzyme pepsin works well in strong acidity, while enzymes in the small intestine require near neutrality. Each one has its own environment, its own balance, finely tuned to its task. Without this tuning, the chemistry of life would falter. As you rest, pH balance is being maintained within you, silently and without effort. Each breath you take adjusts the levels of carbon dioxide in your blood, gently influencing acidity. Each beat of the heart carries buffers that stabilize changes. The kidneys filter fluids, reclaiming what is needed, releasing what is not. Proteins in your cells lend their side chains to the effort, binding and releasing ions to keep conditions steady. This orchestration is constant, unbroken, and unnoticed. Yet it is what allows your body to continue in calm equilibrium. To reflect on pH is to see life sensitivity and resilience side by side. The range for survival is narrow, yet systems exist to hold within that range, layer upon layer of defenses, ensuring that balance is preserved. It is a reminder that life is not about extremes but about harmony, not about rigidity but about adaptability. The chemistry of balance is gentle, quiet, and essential. As you drift deeper into rest, you may imagine this balance within you, like a calm lake that resists ripples, always returning to stillness. Your body is a vessel of quiet regulation, with buffers, lungs, kidneys, and proteins, all contributing to the same purpose: stability. They work silently, tirelessly, and without need for your direction. And as you breathe calmly, you can take comfort in knowing that the balance holds, steady and sure, carrying you through the night. 16. Chemical cycles in nature. Life does not exist in isolation. Every living being, from the smallest microorganism to the largest tree, depends on the environment around it, and in turn, contributes to that environment. The chemistry of life is not confined within cells or bodies, but flows outward, connecting organisms to soil, water, and air. This flow occurs through cycles, great patterns of renewal that move elements and molecules through ecosystems and across the planet. These cycles ensure that the building blocks of life, carbon, nitrogen, oxygen, water, and others are not exhausted but are continually recycled. They are nature's way of keeping balance, of ensuring that life can persist generation after generation. To reflect on chemical cycles is to see the quiet interdependence of all living things, linked by shared chemistry that moves endlessly through the earth. One of the most familiar of these is the carbon cycle. Carbon is the backbone of organic molecules, the framework for carbohydrates, proteins, lipids, and nucleic acids. It is also present in the atmosphere as carbon dioxide, a gas that plants draw in during photosynthesis. Using sunlight, plants transform carbon dioxide and water into glucose and other organic compounds, storing energy in chemical bonds. These compounds become the food that sustains animals, fungi, and microbes. When these organisms respire, they break down the molecules, releasing carbon dioxide back into the air. When they die, decomposers feed on their remains, returning carbon to the soil and releasing gases to the atmosphere. Over longer timescales, carbon can become locked in rocks, sediments, or fossil fuels, only to be released again through volcanic activity or human use. The cycle is vast, connecting breath to tree, soil to sky, ocean to land, and reminding us that the carbon in our bodies is the same carbon that once drifted in the atmosphere or settled in ancient seas. The nitrogen cycle is equally vital. Nitrogen is a key element in amino acids and nucleotides, the building blocks of proteins and DNA. Though nitrogen gas makes up the majority of Earth's atmosphere, most organisms cannot use it in this form. Instead, specialized bacteria perform nitrogen fixation, converting nitrogen gas into ammonia and related compounds that plants can absorb. These compounds enter the food chain as animals eat plants, and they become part of proteins and nucleic acids within their bodies. When organisms excrete waste or die, other bacteria break down the organic nitrogen, returning it to the soil. Some bacteria even convert nitrogen compounds back into gas, releasing it to the atmosphere and completing the cycle. Without this circulation, nitrogen would remain locked in the air, unavailable to life. The nitrogen cycle shows how even the most abundant resource requires transformation, and how microorganisms quietly maintain balance for all living beings. The oxygen cycle, too, sustains life with its rhythm. Oxygen is produced primarily through photosynthesis, when plants and algae release it as a byproduct of capturing sunlight. Animals, fungi, and many bacteria consume this oxygen during respiration, using it to help extract energy from glucose and releasing carbon dioxide in return. This exchange, photosynthesis and respiration, creates a balance in which oxygen is replenished and consumed in harmony. Oxygen also participates in cycles beyond biology, binding to elements in rocks and minerals, or dissolving into oceans and lakes. It is not static but flows through air, water, and earth, always in motion. This cycle illustrates the deep partnership between plants and animals, between producers and consumers, a partnership that has shaped life for billions of years. The water cycle is perhaps the most visible. Water evaporates from oceans, rivers, and lakes, rising into the atmosphere as vapor. It condenses into clouds and falls again as rain or snow, nourishing land and replenishing rivers. Plants draw it up through their roots and release it through transpiration, adding to the atmosphere once more. Animals drink it, use it in their cells, and return it through respiration and waste. Even underground, water flows through aquifers, moving slowly but steadily. This endless cycle ensures that water is available for all life, distributing it across the planet, linking clouds to rivers, plants to oceans, and every living being to the next. Beyond these major cycles, others weave through the fabric of life. The phosphorus cycle moves phosphorus, an essential component of DNA, RNA, and ATP, from rocks into soil and water, where plants absorb it and pass it through food chains. Unlike nitrogen or carbon, phosphorus has no significant gaseous form, so its cycle depends on erosion and geological processes, moving slowly but steadily over time. The sulfur cycle, too, sustains life, as sulfur forms part of certain amino acids and vitamins. Volcanic activity, bacterial metabolism, and the weathering of rocks all contribute to its circulation. These cycles may be less visible, but they are no less crucial, reminding us that every element has its path, every nutrient, its rhythm of renewal. What makes these cycles remarkable is their interconnectedness. The carbon cycle depends on the oxygen cycle, for carbon dioxide and oxygen are exchanged together in respiration and photosynthesis. The nitrogen cycle affects the carbon cycle, since the growth of plants, which draw in carbon, depends on nitrogen availability. The water cycle influences them all, carrying nutrients through soil, rivers, and oceans, and enabling reactions in every living cell. These cycles do not exist separately but overlap, forming a web of chemical motion that sustains ecosystems and connects the biosphere to the atmosphere, lithosphere, and hydrosphere. The cycles are also resilient, adapting to changes and disruptions. When carbon dioxide levels rise, plants may grow faster, drawing in more carbon. When nitrogen is scarce, certain bacteria become more active, fixing more from the atmosphere. When water is limited, organisms adapt, conserving moisture, slowing processes until balance returns. Yet there are limits, and when disruptions exceed the cycle's ability to adapt, ecosystems feel the strain. Still, the very existence of these cycles speaks to the deep resilience of Earth's systems, which have sustained life for billions of years through shifting climates, volcanic eruptions, and continental drift. On a personal scale, these cycles connect us to the wider world. Every breath you take draws oxygen that may have been released by a leaf only hours ago. Every exhale releases carbon dioxide that may be absorbed by a plant tomorrow. The nitrogen in your muscles and DNA once passed through the soil, perhaps through a bean or a grain of wheat. The water in your body may have fallen as rain a year ago, or as snow a century ago, or drifted in the ocean for thousands of years before returning to land. To live is to participate in these cycles, to be part of the quiet flow that binds all beings to Earth. As you rest, the cycles continue. Plants release oxygen into the night air. Microbes in the soil fix nitrogen, unseen yet essential. Water rises from lakes and rivers into the sky, forming clouds that will rain again. Carbon circulates through your breath, out into the atmosphere, into the leaves of trees, and back again in endless rhythm. These cycles are not hurried. They move with patience, with persistence, with the steadiness of processes that have endured for ages. They remind us that life is not only individual but collective, not only momentary but continuous. To reflect on chemical cycles is to find comfort in continuity. The elements that sustain you have been part of countless beings before, and will be part of countless beings after. They are never lost, only transformed, passed from air to soil, from water to life, from one generation to the next. This quiet recycling is the great promise of nature, that life endures through sharing, through connection, through cycles that renew themselves endlessly. As you sink deeper into calm, you may imagine yourself as part of these cycles, your breath, your body, your being linked to the wider earth. The carbon, nitrogen, oxygen, and water within you are not yours alone, but part of a greater flow. They will move on, as they always have, and in that movement, life will continue. These are the chemical cycles of nature, the silent, steady rhythms that sustain the world and all who dwell within it. 17. Trace elements and their subtle roles. When we think of the chemistry of life, our minds often turn to the familiar elements: carbon, hydrogen, oxygen, and nitrogen. These make up the majority of living matter and form the backbone of proteins, carbohydrates, lipids, and nucleic acids. But life depends not only on these abundant players. Scattered throughout biology are elements required in much smaller amounts, sometimes only a few milligrams in the entire body, sometimes even less. These are the trace elements. They may be subtle, their presence almost hidden, but their roles are profound. Without them, enzymes would falter, proteins would lose their function, and balance would collapse. Trace elements remind us that even the smallest contributions can sustain the largest systems, that life is built not only on abundance but also on quiet precision. Iron is perhaps the best known of the trace elements. Though present in only modest amounts, it carries oxygen throughout the body. Within hemoglobin, the protein found in red blood cells, iron binds to oxygen molecules in the lungs and releases them in tissues where they are needed. Each hemoglobin molecule contains 4 iron atoms, and without them, oxygen transport would fail. Iron is also found in myoglobin, a protein that stores oxygen in muscles, ensuring a steady supply during activity. Beyond oxygen transport, iron participates in electron transfer reactions in mitochondria, helping generate ATP. The importance of this metal cannot be overstated, yet it is needed in only small amounts. Too little, and the body struggles with anemia, leaving tissues starved of oxygen. Too much, and iron becomes toxic, generating harmful free radicals. Iron illustrates the delicate balance of trace elements, essential yet dangerous in excess. Zinc is another trace element with wide-reaching influence. Found in hundreds of enzymes, zinc acts as a cofactor, enabling reactions to proceed that would otherwise be impossible. It helps enzymes stabilize structures, bind to molecules, and carry out chemical transformations. Zinc is also crucial for the function of transcription factors, proteins that regulate gene expression by binding to DNA. Without zinc, growth, repair, and immunity falter. It supports the development of the immune system, assists in wound healing, and plays a role in taste and smell. Even though zinc is needed in minute quantities, its absence can weaken the body's defenses and impair its ability to regenerate. Copper, like iron, participates in electron transfer. It is found in enzymes that help generate energy in mitochondria and in enzymes that defend against oxidative stress. Copper also plays a role in forming connective tissues, as it is required for the cross-linking of collagen and elastin. In the nervous system, copper contributes to the production of neurotransmitters, ensuring that signals can flow smoothly from one neuron to another. Its reddish sheen and pure form belies the subtlety of its presence in biology, where even a trace supports vital functions. Manganese is a quiet helper in many enzymatic processes, It contributes to the metabolism of carbohydrates, amino acids, and cholesterol. It also forms part of enzymes that protect against oxidative stress, ensuring that reactive oxygen species do not damage cells. Plants depend on manganese for photosynthesis, where it participates in splitting water molecules to release oxygen. Without manganese, the flow of energy in both plants and animals would be disrupted, reminding us again that even elements present in tiny amounts are indispensable to the whole. Selenium, though required in microgram quantities, is vital for defense. Incorporated into selenoproteins, it plays a role in antioxidant systems that protect cells from damage. Selenium also contributes to thyroid function, helping regulate metabolism and growth. Its presence in soil influences its availability in plants and thus in animals, showing how trace elements connect ecosystems together. Regions with selenium-poor soils may see deficiencies ripple through food chains, while regions with too much may face toxicity. This duality is common among trace elements. Both deficiency and excess can be harmful, underscoring the need for balance. Iodine is best known for its role in the thyroid gland. It is a key component of thyroid hormones, which regulate metabolism, development, and growth. Without sufficient iodine, the thyroid cannot produce enough hormone, leading to disorders that affect the whole body. Iodine deficiency has been addressed in many parts of the world by fortifying salt, a reminder of how awareness of trace elements has improved human health. Like other trace elements, iodine works quietly, its effects unseen until balance is lost. Cobalt, though needed in tiny amounts, is essential as part of vitamin B12. This vitamin plays roles in DNA synthesis, red blood cell formation, and the function of the nervous system. Without cobalt, the vitamin cannot function, and deficiencies can lead to anemia and neurological issues. Here again we see the subtlety of trace elements. Cobalt itself is not abundant in tissues, but without it, a crucial vitamin loses its power. Molybdenum is another trace element required only in minute quantities. It is part of enzymes that help break down nitrogen-containing compounds, sulfur compounds, and even certain toxins. Its role in detoxification and metabolism keeps the body's chemistry balanced. Without it, harmful substances could accumulate, disrupting life's quiet equilibrium. These elements, though few in amount, show the principle that life depends not on size or abundance, but on function. Each trace element occupies a unique niche, supporting enzymes, stabilizing proteins, or regulating balance. Their roles are not interchangeable. Iron cannot replace zinc, nor can copper take the place of iodine. Each one carries a specific responsibility, like a small cog in a vast machine. Remove the cog, and the whole system falters. The story of trace elements also connects us to Earth itself. These elements come from rocks, soil, and water. They are taken up by plants, passed into animals, and eventually return to the ground through decay. Their availability depends on geology, climate, and ecosystems, linking life to the planet's structure. A deficiency in soil can ripple upward through plants, animals, and humans, showing that the chemistry of life is inseparable from the chemistry of Earth. In this way, trace elements remind us that our bodies are not apart from nature but part of it, built from the minerals beneath our feet, and sustained by the cycles that renew them. As you rest, trace elements are at work within you. Iron carries oxygen to your tissues, zinc supports enzymes, copper guides energy flow, and selenium protects your cells from harm. These elements are quiet, their amounts too small to notice, yet their influence reaches into every part of your being. They are always present, always steady, fulfilling their roles without pause. To reflect on trace elements is to see the value of subtlety. Life does not rely only on what is abundant, but also on what is scarce. The smallest contributions can make the greatest difference. The quietest roles can sustain the largest systems. This is the lesson of trace elements, that life is built on balance, on harmony, on the cooperation of both the great and the small. As you drift deeper into calm, you may imagine these elements within you, tiny sparks of function scattered through your body, each one supporting the flow of life. They are part of you, yet they also connect you to soil, to water, to the earth itself. They remind you that even the smallest parts matter, that even the faintest presence can be essential. These are the trace elements, subtle and profound, the quiet guardians of life's chemistry. 18. Origins of biochemistry on the early Earth. To understand life as it is today, it is natural to look back to its beginnings. The chemistry that flows through cells, the molecules that carry energy, the elements that sustain us. All of these have roots deep in the history of the Earth, long before the first plants reached toward the sun, long before animals swam in seas or walked on land. The building blocks of life were forming in the quiet chemistry of the early planet. The origins of biochemistry are the story of how a barren Earth gave rise to molecules that could replicate, metabolize, and eventually evolve into living beings. It is a story not of sudden leaps but of gradual steps, each one guided by the laws of chemistry and the patience of time. The Earth formed around 4.5 billion years ago, a young planet marked by heat, volcanic activity, and bombardment from space. The atmosphere then was different from what we know today. Instead of abundant oxygen, it likely contained gases such as methane, ammonia, water vapor, hydrogen, and carbon dioxide. Oceans were forming as the planet cooled, providing vast reservoirs where chemical reactions could unfold. In this environment, simple molecules interacted, driven by energy from lightning, ultraviolet radiation, and geothermal heat. These conditions, though harsh, were fertile ground for the emergence of biochemistry. One of the most famous ideas about this early chemistry is the concept of the primordial soup. In this view, the oceans contained a mix of simple molecules that, under the influence of energy, formed more complex organic compounds. Experiments in the 20th century, such as the Miller-Urey experiment, showed that when gases thought to resemble the early atmosphere were exposed to sparks of electricity, amino acids and other organic molecules could indeed form. These findings suggested that the basic ingredients of life could arise spontaneously under the right conditions, providing the first steps toward biochemistry. Amino acids, simple sugars, nucleotides, and lipids, these building blocks may have been created on Earth, or they may have arrived from space. Meteorites have been found to contain amino acids and other organic molecules, hinting that the seeds of life could have been delivered from beyond our planet. Whether formed here or brought from afar, these molecules began to gather, accumulate, and interact in the waters and soils of the early Earth. The next challenge was organization. Life depends not only on molecules but also on their arrangement. One hypothesis is that lipids, with their ability to form membranes spontaneously, played a key role. When fatty acids are placed in water, they naturally assemble into vesicles, tiny bubbles with a boundary between inside and outside. These simple compartments could have provided a sheltered space, concentrating molecules and protecting them from the outside environment. Within these vesicles, reactions could proceed more efficiently, laying the groundwork for primitive cells. Nucleic acids may also have emerged in these early environments. RNA, in particular, is thought to have been central to the first biochemistry. Unlike DNA, which primarily stores information, RNA can both store genetic instructions and act as a catalyst for reactions. This dual ability has led to the RNA world hypothesis, which suggests that life may have begun with RNA molecules that could replicate themselves and catalyze chemical processes. Over time, these RNA molecules may have given rise to proteins for greater catalytic diversity and to DNA for more stable information storage. Minerals on the early Earth may have assisted in these processes. Clay surfaces, for instance, can attract and concentrate organic molecules, aligning them in ways that encourage bond formation. Metal ions from rocks may have acted as cofactors, guiding reactions toward useful products. Hydrothermal vents on the ocean floor, where hot water rich in minerals emerges, may have provided both energy and the right conditions for chemistry to advance. These vents, with their compartments and chemical gradients, resemble natural laboratories, where biochemistry could have taken its first steps. Another important aspect of early biochemistry is energy. Life requires a constant flow of energy, and on the early Earth, multiple sources were available. Sunlight provided abundant radiation, some of which penetrated the atmosphere to reach the surface. Lightning offered bursts of electrical energy. Geothermal activity released heat and chemical gradients from the planet's interior. Primitive systems may have learned to harness these sources, capturing energy in simple molecules such as ATP or in proton gradients across membranes. In this way, the flow of energy that sustains life today may have its roots in ancient processes driven by Earth's natural forces. As molecules grew more complex, interactions became more organized. Chains of amino acids may have begun to fold into simple proteins, offering new catalytic abilities. Nucleotides linked into strands of RNA, capable of carrying information and assisting in their own replication. Lipid membranes enclosed these processes, creating stable environments where chemistry could persist and adapt. With each step, the system grew more lifelike, until at some point, it crossed the boundary into what we now call life. That boundary is not sharp, but gradual, as chemistry slowly acquired the qualities of biology: replication, metabolism, and evolution. The emergence of metabolism was a crucial milestone. Primitive systems began to capture energy from their surroundings and use it to build new molecules. Instead of relying only on the environment for supplies, they learned to sustain themselves. Pathways resembling glycolysis or fermentation may have appeared, guiding the flow of carbon and energy in controlled ways. The ability to harness and regulate reactions was a turning point, transforming passive chemistry into active biochemistry. From these beginnings, life diversified. Some systems may have developed the ability to capture sunlight, giving rise to early photosynthesis. This process eventually transformed the planet, releasing oxygen into the atmosphere and reshaping Earth's chemistry. Others thrived near hydrothermal vents, using chemical gradients as their energy source. The diversity of strategies shows that life did not emerge in a single form, but explored many possibilities, guided by chemistry's flexibility and Earth's varied environments. The origins of biochemistry are not only a story of Earth, but also of connection to the cosmos. The elements that make up life — carbon, hydrogen, oxygen, nitrogen, phosphorus, sulfur — were forged in the hearts of stars and scattered through space in supernovae. The molecules that seeded the early Earth may have formed in interstellar clouds or on comets. In this sense, the chemistry that became biology is part of a much larger story, one that began long before Earth existed. To reflect on this is to see that life is not isolated, but woven into the fabric of the universe. As you rest, you carry within you the products of this ancient chemistry: the DNA in your cells, the ATP that fuels your muscles, the proteins that sustain your tissues. All are descendants of processes that began billions of years ago in Earth's oceans and rocks. The molecules that support you now are built on patterns that stretch back to the dawn of life, refined by time but rooted in the same quiet chemistry. To reflect on the origins of biochemistry is to recognize the patience of nature. Life did not appear suddenly, but through slow accumulation, through countless small steps, guided by the steady laws of chemistry and the enduring cycles of Earth. It is a reminder that complexity can grow from simplicity, that persistence can create possibility, and that life itself is the outcome of patient chemistry unfolding over time. As you drift into calm, you may imagine the early Earth, with its oceans, its storms, its heat and minerals. You may picture simple molecules gathering, forming vesicles, linking into chains, capturing energy. From those beginnings came the flow of chemistry that now sustains you. You are part of that story, a continuation of processes that began long ago. The origins of biochemistry are not only in the past but within you, present in every cell, every breath, every heartbeat, a quiet echo of Earth's earliest days. 19. The harmony of biochemistry and life's continuity. Biochemistry, in its essence, is a symphony. Each molecule, each reaction, each cycle plays its part, contributing to the larger music of life. When we look closely, we see not isolated processes, but harmony interactions that balance one another, rhythms that repeat yet adapt, themes that echo across generations. This harmony is what allows life to persist, not only in a single moment, but across time, through change, through challenge, through continuity. To reflect on the harmony of biochemistry is to recognize that life is not only chemistry, but chemistry in balance. Chemistry arranged into patterns that sustain and renew. The harmony begins with molecules themselves. Proteins, carbohydrates, lipids, and nucleic acids are distinct, yet they interact seamlessly. Proteins fold into enzymes that build and break molecules. Carbohydrates provide fuel. Lipids form barriers. Nucleic acids carry instructions. None of these macromolecules exists in isolation. Each depends on the others. DNA encodes proteins. Proteins build and repair DNA. Carbohydrates fuel the process. Lipids protect the structures where it unfolds, like the sections of an orchestra's strings. Winds, percussion, they are different in form, but united in purpose. Together they create the living score. This harmony extends into metabolism. Catabolism and anabolism are opposite in direction but complementary in function. One breaks down, releasing energy. The other builds up, consuming it. The ATP produced in catabolic reactions is spent in anabolic ones, completing the circle. The waste from one pathway becomes the raw material for another. Even within a single cell, dozens of pathways flow together, each one adjusted by enzymes, each one influenced by signals, each one contributing to the whole. This orchestration ensures that nothing is wasted, that energy flows smoothly, that life remains steady. The harmony also lies in regulation. Too much activity in one direction can unbalance the system, so feedback loops exist to restore equilibrium. When glucose is plentiful, pathways for storage are activated, converting it to glycogen or fat. When glucose is scarce, pathways for release are triggered, breaking down reserves to provide fuel. Hormones carry these signals across the body, coordinating cells and tissues so that the whole organism acts in unison. Insulin lowers blood sugar by promoting storage. Glucagon raises it by promoting release. Each hormone counters the other, not in conflict, but in cooperation, maintaining balance. On a larger scale, the harmony of biochemistry connects organisms to their environment. The carbon dioxide you exhale becomes fuel for plants. The oxygen they release sustains your breath. Nitrogen cycles from atmosphere to soil to organism and back. Each step guided by enzymes in microbes, plants, and animals. Water evaporates, condenses, falls, and returns, nourishing every level of life. These cycles are not separate from biochemistry but extensions of it, the same chemistry playing out in ecosystems as in cells. The harmony of life is not only within organisms but across the planet, linking air, water, soil, and living beings in one grand process. Continuity emerges from this harmony. The molecules of life are rebuilt again and again, yet the patterns endure. DNA is copied each time a cell divides, ensuring that instructions are preserved. Proteins are broken down and remade, yet their functions continue. Energy flows through ATP, always spent and always regenerated. Cells die, tissues renew, organisms pass, yet life persists, because chemistry continues its cycles without end. Continuity is not the survival of individual molecules, but the persistence of patterns. The unbroken music of biochemistry. This continuity is visible across generations. The DNA in your cells carries instructions that trace back through countless ancestors. Refined but preserved, the proteins in your body fold the same way they folded in ancient organisms, guided by the same forces. The ATP that fuels your cells is the same form of energy used by the first bacteria billions of years ago. Even the cycles of carbon, nitrogen, and water have turned endlessly, linking you to the earliest life forms. Continuity means that life is not a series of isolated events, but a flowing river of chemistry, with each generation a moment in its course. Harmony also allows for change. Biochemistry is stable, but not rigid. Mutations in DNA create variations, some harmful, some neutral, some beneficial. Enzymes evolve new functions. Pathways adapt to new environments. Organisms develop new strategies for survival. The harmony of chemistry is flexible, able to absorb change without losing balance. This adaptability is what allows life not only to continue, but to evolve, to expand into new forms, to explore new possibilities. Continuity, then, is not sameness, but persistence through variation. The harmony of biochemistry can also be seen in its efficiency. Reactions are not random, but ordered. Molecules are not wasted, but recycled. Energy is not squandered, but managed with precision. The body can shift seamlessly between fuels, using carbohydrates, fats, or proteins depending on availability. Cells can sense their energy state and adjust metabolism accordingly. Enzymes work only when needed, and feedback ensures that pathways do not overproduce. This efficiency is a form of harmony, a quiet balance that conserves resources and sustains life. As you rest, this harmony flows within you. Your heart beats in rhythm, fueled by ATP, generated from glucose and oxygen. Your brain fires signals, maintained by ion gradients across membranes. Your cells build proteins, guided by DNA, assisted by RNA, catalyzed by enzymes. At the same time, waste is cleared, balance restored, and energy renewed. You are not aware of it, yet this harmony sustains you every moment, carrying you from one breath to the next, from wakefulness into sleep. To reflect on the harmony of biochemistry is to see the quiet beauty of life. It is not chaotic, nor is it mechanical, but something in between, ordered yet flexible, precise yet adaptable. It is a pattern that repeats but also changes, a melody that continues but also evolves. This harmony is what allows life to persist, not just in one organism, but across the whole of existence, through time and across space. As you drift deeper into calm, You may imagine this harmony within you, like music playing softly, always steady, always present. You are part of that music, your body sustained by the flow of biochemistry, your life a note in the larger song of Earth. And though individual molecules come and go, though cells and organisms rise and fall, the music continues, unbroken, weaving continuity from chemistry, balance from complexity, life from matter. 20. Biochemistry as a universal language of life. Across the immense diversity of life, from the simplest bacterium to the most complex human, there exists a common thread, the chemistry that sustains them. Despite the variety of shapes, behaviors, and habitats, all living beings speak the same chemical language. They use the same nucleotides to write their genetic code, the same amino acids to build proteins, the same ATP to carry energy, and the same cycles to process carbon, nitrogen, and water. This shared foundation is the universal language of biochemistry, a language written not in words but in molecules, not in sounds but in reactions. It is a language so consistent that it unites all organisms into a single family, stretching back billions of years to a common origin. This universality begins with DNA and RNA, the molecules of information. Every organism stores instructions in the same code, 4 bases arranged in sequences that specify proteins. Adenine pairs with thymine, guanine pairs with cytosine, and in RNA, uracil takes the place of thymine. From bacteria to humans, the code is the same, the alphabet unchanged. Even the way the code is read, 3 bases at a time forming codons, is shared. The codon AUG signals the start of a protein, while UAA, UAG, and UGA mark the end. This genetic code is universal, so much so that scientists can take a gene from one organism and place it in another, and it will still be read and expressed. The fact that all life shares this language speaks to its deep ancestry and to the power of chemistry to unify. Proteins, too, are built from the same set of 20 amino acids in nearly every organism. These amino acids are linked into chains by peptide bonds, folding into shapes, guided by the same principles of chemistry. Hydrophobic side chains tucked inside, charged ones exposed, hydrogen bonds stabilizing turns and sheets. A protein in a human and a protein in a bacterium may serve different functions, but the rules that shape them are identical. This consistency allows for endless diversity of form and function while preserving a common foundation. The language of proteins is universal not only in its alphabet of amino acids but in its grammar of folding and catalysis. The energy of life is also carried in the same form across all beings. ATP, whether in a leaf capturing sunlight, a muscle contracting during motion, or a bacterium dividing in soil, ATP serves as the currency of energy. It is generated through pathways that are also universal. Glycolysis, the citric acid cycle. These processes, discovered early in evolution, have been preserved because they are efficient, reliable, and flexible. The universality of ATP shows that life has not reinvented its currency, but is held to a standard that connects every organism across time and space. Metabolic pathways reveal this unity even more clearly. The steps of glycolysis, breaking glucose into pyruvate, are found in nearly every living cell. The citric acid cycle, processing carbon fragments and releasing electrons, is equally widespread. Even in organisms that live in extreme environments, these pathways remain, sometimes modified but still recognizable. This suggests that the language of metabolism was established early and has endured because it works. To study metabolism in a bacterium is to glimpse the same patterns that unfold in human cells. separated by billions of years, yet speaking the same biochemical tongue. The universality of biochemistry also extends to the cycles that sustain ecosystems. Plants capture carbon dioxide and water, turning them into sugars and oxygen. Animals consume the sugars and release carbon dioxide back. Nitrogen is fixed by bacteria, taken up by plants, passed through food chains, and returned to the atmosphere. Water evaporates, condenses, falls, and flows. These cycles are not the work of one species or one kingdom, but of all life together, each organism playing its role in a shared chemical rhythm. The cycles are like conversations across the biosphere, exchanges in the universal language of chemistry. Even the smallest details reveal this unity. Enzymes in vastly different organisms often share similar structures, their active sites shaped in ways that transcend species. Hemoglobin in humans and hemoglobin-like molecules in invertebrates perform the same task, carrying oxygen. Photosynthetic pigments in plants and cyanobacteria absorb light in overlapping spectra. These parallels show that life does not use isolated dialects of chemistry, but draws from the same vocabulary, the same set of tools. This universal language allows us to see kinship where the surface shows difference. A tree, a fish, a bacterium, and a human may appear unrelated, yet their biochemistry tells another story. The DNA that guides their growth is written in the same code. The proteins that carry out their reactions are built from the same amino acids. The ATP that fuels their cells is the same molecule. The metabolic cycles that sustain them are variations on the same theme. This kinship reminds us that all life is related. All life shares a common ancestry. All life is part of one continuous story. The universality of biochemistry also opens the possibility of connection beyond Earth. If life exists elsewhere in the universe, it may be based on similar principles. For the laws of chemistry are not confined to Earth. Carbon's versatility, water's properties, the stability of nucleotides and amino acids all are universal features of matter. While alien life may look different, it may still speak a language recognizable to us, a biochemical tongue shaped by the same physical laws. To understand the universal language of life on Earth is to prepare for the possibility of hearing it elsewhere, in distant worlds. What is most striking about this language is its balance between simplicity and complexity. The alphabet is small, 4 nucleotides, 20 amino acids, a handful of energy carriers, yet from this simplicity arises vast diversity. Just as human languages use limited letters to create endless words and meanings, biochemistry uses limited molecules to create endless forms and functions. This is the power of universality. It allows complexity to grow from a shared foundation, ensuring both continuity and creativity. As you rest, this universal language is being spoken within you. DNA is being read into RNA. RNA is guiding the making of proteins. Proteins are catalyzing reactions. ATP is being spent and replenished. Molecules are flowing through cycles. These processes are not unique to you but shared with every living being. From the smallest microbe in the soil to the largest tree in a forest, you are part of this language. Your life a continuation of the conversation that began billions of years ago. To reflect on biochemistry as a universal language is to find unity in diversity. It is to see that beneath the variety of life, there is a common script, a shared song, a language spoken by every cell, every organism, every ecosystem. It is a language without borders, without time, without interruption. It is the language of life itself, steady, patient, and enduring. As you drift deeper into calm, you may imagine this language flowing within you and around you, connecting you to all living things. Each breath, each heartbeat, each quiet reaction is a word in that language, a note in that song. The universal language of biochemistry binds you to the earth, to the past, to the future, and to the whole of life. It is a reminder that you are not alone, but part of a greater story, one written in molecules, spoken in reactions, and sung in the harmony of existence.